Hello, 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 everybody. I am Phi the Neutrophil. Hello, welcome to the stream. It's been a little bit of time, everybody. How is everybody doing on this fine Wednesday afternoon? At least it is Wednesday here. Today, we are going to be talking about the brain, a very complicated, complex organ, one that I am fully unprepared to talk about on my own, despite having had training in neuroimmunology, although we'll get into a little bit about why that background may have actually hampered me from understanding this paper in its entirety. But to help me on this process, we have here a real psychiatrist. Real psychiatrist, please introduce yourself.
Hi, everybody. I'm almost a real psychiatrist. My name's Nils Wendell. Yeah, I am in my 4th year of training at Tufts. So yeah, like I said, almost a real psychiatrist. They don't let me out of the hospital unsupervised yet, but soon.
Soon you will have yet another license, because you already have a license to practice medicine, but you'll have like a double full adult Omega empowered license to do brain stuff out in the open, which is very exciting.
Yes, exactly.
So while people are filing in, I don't often get the opportunity to have someone in a health-related field, although I just had Psychin come on. But if you want to just get us started with a little bit about what it's like being a psychiatrist, I'm sure some of my audience might be curious.
Yeah. So being a psychiatrist is a lot of fun. I love it. I went to medical school to go into psychiatry. I kind of figured that out relatively early on when I went to college. You know, I was a bio major the whole time, but I was like, what am I going to do with a biology degree? And I was like, yeah, I guess I'll go into medicine. But what kind of medicine am I going to do? And psychiatry always just kind of seemed to be the right thing for me. Psychiatry is very varied. There are a lot of different things that you can do. In psychiatry, part of our training, we get exposed to a lot of different stuff. We do some inpatient work. So those are is fancy way of talking about the people who are kind of the sickest of the sick, like people who need to be in lock supervised settings because they might hurt themselves or they might hurt somebody else. We will go and help our colleagues out on the medical floors if they have a question that they think that we might be able to help with. Sometimes, you know, could this medical thing be actually a psychiatric thing? Or, you know, we need help managing this person's addiction or whatever else. You know, there's emergency psychiatry where we see people kind of in crisis and we figure out, you know, what level of care is appropriate for them. And then what I've been doing lately is outpatient psychiatry, which I think is probably what a lot of people think of when they think of psychiatry is, you know, just like going to a doctor, like any other doctor outside of a hospital for checkups and, you know, therapy and so on.
I think what would be a nice segue into talking about the paper that we have is, so we got together and we kind of brainstormed what would be appropriate for us to talk about. And so this topic was one that you had suggested, and I don't want to spoil too much about what's coming up in your presentation, but could you introduce the audience a little bit as to how this might tie into your practice of psychiatry, or why you might be interested in this topic of predictive processing without getting into too much detail? Just a teaser for what's to come.
Yeah, I will do my best. So for the audience, psychiatry, you know, like from the outside might look like a very put together field of medicine, like a lot of the other fields of medicine. You know, we have our diagnoses, we have schizophrenia, we have major depressive disorder, we have generalized anxiety disorder, so on and so forth. But our diagnoses are kind of made up. We made up in the sense, like, we don't have a good explanation for why a lot of these things happen, right? Like, we can't tell you, you know, if someone has heart failure, a cardiologist can tell you why someone's having heart failure. It's, oh, there's a piece of the muscle that's dead. Oh, you have a disease which causes your heart muscle to break down, and so your heart isn't pumping as well. We don't really have that for psychiatric diseases. We have a collection of symptoms, and the cause of those symptoms we don't really know.
So we have a comment here from Izzy saying that the DSM, the criteria, can be very vague too. Do you think that plays a role in this sort of mechanistic unknown?
Yeah, well, right, exactly. So the DSM is, so for those of you who don't know, that's, I think, the Diagnostic and Statistics Manual. We are currently on its 5th edition. And it's really our illnesses, our disorders, are collections of symptoms. And that's kind of about as deep as we go. So the cool thing about predictive processing to me is that it starts to offer an explanation for what is going wrong in people with various disorders by giving us an explanation, more of an explanation about how the brain is set up, and how it works in a way that kind of allows us to maybe start thinking about these diagnoses in a unified way, as opposed to like, oh, you know, um, the brain of with someone with schizophrenia is just worlds and worlds apart from someone with depression or anxiety. Like maybe that's not so true, if we think about it from a predictive processing point of view, and that's very exciting.
Yeah, so as someone who lives in the realm of molecules and cells, things that I can imagine, the brain is so extremely complex that I've had a really tough time with this paper due to how much abstraction is the wrong term, but like if we can get a handle on how the brain works mechanistically, it would be very exciting because that's the kind of thing that I like to study on my own. So I'm really excited to get into this discussion of what might be underlying some psychiatric disorders, as well as just how the brain could be functioning at a molecular level, which I'm super hyped for. All right, without further ado, we are actually going to have this first part of the presentation done by Dr. Wendell himself, because I am wholly unequipped to talk about this at a deeper level than the paper. So, Dr. Wendell, let's get started.
Yeah. All right. So I'm, to be honest, Fi, not that much more equipped than you are, but we'll do our best to muddle through it. So who am I? I've already talked about this a little bit, but for the people who filed in a little bit later, my name's Nils Wendell. I'm a 4th-year psychiatry resident at Tufts. My background in this stuff. So I have my bachelor's in biology, concentration in neurobiology and behavior. So I do have a bit of background about this, but it's been a while, since this is the first time in a long time I've really sat down with a really in-depth kind of, neurobio paper at this level for a while, which was fun to do. It was nice to refresh my memory about what it was like to be back in college. I also got my minors in cognitive science and psychology too, so you can kind of see where my interests lie from Cornell. Disclaimer, I'm not an expert in predictive processing. I don't have a PhD. I have not spent years of my life studying this stuff. I just think it's really cool. I think it's something that's worth talking about. I think it's relevant to my field and to my job as a psychiatrist. So just keep that in mind as we go through this. This is a really complex topic. I'm going to get stuff wrong. Fi's going to get stuff wrong.
I'm definitely going to get stuff wrong.
Yeah, I'm going to act like I know stuff when in reality, sometimes I don't. I'm going to try and be transparent about what I do and I don't know, and what I am confident on, and when I'm giving just a guess. But I just want you guys to be aware, because I don't want to give any, false pretense about me being an expert in this stuff. I'm really not. I just think it's fascinating.
All right, let's go. Let's get to appreciating what this is all about. All right.
Yeah. All right. So a lot of this interest came from this book called Surfing Uncertainty. If any of this piques your interest, highly recommend you go pick up a copy of it. It's not that expensive. It will blow your mind. It has blown my mind multiple times. I'm still not all the way through it. Also comes with a strong recommendation for my cat, Meepo. And what more do you need, honestly? All right, so let's talk about it. So at the core of this, of being a psychiatrist, of doing neurobiology, neuroimmunology, whatever, there's this question of like, how does a brain do brain things? And you can define brain things as widely as you like or as narrowly as you like. But we need some ground rules, right? So let's just assume we're working from a monist perspective. No Cartesian dualism, sorry. For those of you not familiar with that kind of idea, Cartesian dualism is the idea that there is a brain, but then there is also a soul. And I'm going to say no souls allowed, at least for this conversation. I'm not going to get into the metaphysics So we're going to keep it, I'd say simple, but this isn't simple. We're going to keep it as simple as we can, right?
Yeah.
So for a while, and I would say this is still the dominant hypothesis right now about how the brain does brain things, is this bottom-up feed-forward model. Our brain takes in inputs, some sort of sensory information from our sense organs, right? We got a nose, we got eyes, we have, billions of sensory receptors, billions, millions, all across our body. And then all of that information, all of it is fed forward through our brain and synthesized into more and more complex forms. And at the end, we get consciousness and our experience of being, right? And some people thought about this for like many, many, many decades and thought, there are some problems here. This is really energetically expensive and inefficient, right? Like we have to take all of that information in all of the time. Aren't there parts of the world or of our world and our sense data that like aren't that important or that we don't need to refresh all the time? Those of you who know a little bit about computer science, there is a lot of kind of computer science like thinking in this, hey, wait a moment minute, where if you know how compression algorithms work, you're not updating, information that's redundant when you update, say, like a frame of a video. This is, part of how YouTube and Twitch save data, is they're not updating literally everything all the time. And so people said, like, why doesn't our brain do that? It probably does, because Mother Nature does not, she's a spendthrift, right? Or she's very miserly with the amount of energy she's willing to spend for things, right? And the other part of this is that this whole feed-forward model doesn't provide a good explanation as to how a brain organizes itself in the first place. It's just kind of like these systems are thought to spring out of thin air. They're all set up during development and are genetically coded for, and that's kind of it. And that's a little bit confusing. How you could so intricately code for all of these very, very complicated systems that kind of have to grow up together side by side. So enter predictive processing. So predictive processing says, hey, wait a moment, let's go back to basics, right? What at the most basic level are our brains working with? And the answer is energetic stimuli. All our brains have are these little spikes of energy that flow through neurons, the action potentials. And then something happens, and then out at the other end, we get human consciousness, right? And so predictive processing is like, what the hell is going on here? That our brains, like, all they have is this information. And then we get to this much more complex picture. But let's think about like how a system might be set up to take in that information and turn it into something useful, right? And so this question, how might a brain learn to do that? And the answer that predictive processing gives is that we do that by creating a system whose job is to accurately predict the incoming or upwards flow of information, right? So the way that you might think about this in a really simple sense, and I'll try to explain this as best as I can, and Fi, let me know if you feel like I'm glossing over some of the mechanics a little too briefly here. But you can think of it in this really simple kind of neuron schema here. So here you have a sensory neuron, right? And sensory neurons will say they're just delivering some information. And they're delivering this information to an error detector of some sort. And this error detector is passing that information up in turn to a top-down predictor. So the top-down predictor's job here, right, is to cancel out this incoming sensory information. And so it does this through an interneuron here, which uses a neurotransmitter called GABA, which is the major inhibitory neurotransmitter in the brain, to dampen the activity of this error detector. So this is a really simple way to maybe think about this idea of how you predict bottom-up flow, right? Because what needs to happen is this top-down predictor needs to take the information it's getting from this neuron here and use it to send a signal that's going to cancel out any further information from coming from this cell.
So I think I have a question about this model. And I understand that
Sure.
because it is a model that it necessarily has to be quite simple. But this top-down predictor, just to help me and the audience kind of visualize or imagine what this top-down predictor is, could you say that this is some cell population, for example, in the cortex, like something, because the way that I was imagining it is that you have these sensory neurons, these quote unquote dumb neurons, whose only job it is to take, smell, touch, heat, whatever, and then send that signal up to the thinking brain, like quote unquote the smart neurons. Is that kind of an okay way to think about that?
Let's see. I think,
Or do you think it's a little more complicated than that and I'm oversimplifying it?
I think it's a little bit more complicated than that, Phi. What I want people to kind of get in their heads is that this really simple schema exists at all layers of our brain, from the most complicated parts in the forward part of our brain all the way down to, like, before you even get into the brain, like the neurons that sit in our retina and aggregate data from our photoreceptors. Like, they all have a system like this too. And so there's adjustment at every kind of level about what information is going to get fed forward into the system.
Okay, so that's really helpful. So this isn't a circuit that spans the entire nervous system, as in it's more local than that. Like, this is a potentially fundamental way that neurons talk to each other in any location.
Exactly.
Okay,
right. That's
okay.
the idea of predictive processing, is that this kind of structure, and it's much more complicated than this with many more neurons and, so on and so forth. But this is kind of the gist, is this sort of network.
Aeon has an interesting question. So this doesn't interact with like breathing or heartbeat nerves. I don't think we talk about the autonomic nervous system in this paper at all. And I don't think that's really in the scope of what we both read, but if you had to kind of hypothesize, like, that's an interesting question, because that does have input, but I don't necessarily know if those inputs are sensory. So do you have any thoughts on that?
Yeah, that's a fascinating question. I don't know, actually, the answer to that question. I have some ideas. It does seem intuitively different to me, because it's not something that we necessarily have the sense that it's kind of under control. Like, we don't have any sort of action to perform on modulating those things. But nature likes to reuse anything it can reuse, and this circuitry is so simple that I suspect that it is involved, just in ways that are kind of hard for us to get at, to think about laterally from what you and I are talking about, Phi. So my answer really is, that's a long-winded way of saying I don't know, but great question.
Yeah, great question. Awesome. All right, cool.
All right, so this next slide is just kind of a more simplified, less circuitry sort of system way to think about predictive processing, where, again, and this kind of hammers home this idea that at every level, this interplay between the unpredicted input that we get, the residuals, the errors, those are what is being fed forward up in the processing hierarchy. And what's being sent back down are these predictions to try and cancel out or explain or develop hypotheses for what sensory information we are currently getting. So the lower you get, these signals are very spatially and temporally precise. So temporally, like time-wise, right? When exactly they're happening. They're in the moment sort of pieces of information. And then as you get higher and higher, these predictions get increasingly abstract, right? Thinking about the future, thinking about information from the past, using abstractions like language to help process incoming information, that sort of stuff.
And so just to stay on that figure for a sec, because it's an interesting one. So I don't really know too much about machine learning, but it seems like there is an aspect of you take in like test data and then you auto correct at some point, and this process is continual, like getting the training data, honing the model, et cetera, which I guess is kind of a silly, maybe obvious way that we also learned. We are machines that do learn, but this kind of this interplay I think is really interesting. The fact that it happens sort of simultaneously, at least that's what it seems like from this figure.
Yeah, right, exactly. Like you have adjustments going on simultaneously at every moment, in every context, at every level of your processing hierarchy. It's not just confined to one. Obviously, you're going to have more abrupt changes in particular, flows of information, depending on what you're doing, what's going on, the environment you're in, all that sort of stuff. But yeah.
Very cool.
Exactly. All right. So, to sum all this stuff up, this idea of predictive processing is really nice. It's really nice for a lot of different reasons, but here are a couple. So one, if you set up a system this way, you have your training data available to you, and you can check your work against it really, really quickly, right? Because your training data is the sense data that you're getting in. And if your model isn't accurately predicting the sensory data that it's getting from your eyes, your ears, your nose, your tongue, well, you know what the error is. You have it right in front of you, and your model can iterate on itself very fast. The other part is that this is just way more efficient. Like I've been, mentioning ad nauseum, I'm sure at this point, you can just pass the error forward. You don't have to pass forward all of your sensory data. Just pass forward the stuff that you didn't expect. And incidentally, the stuff that you didn't expect is the stuff that you're going to need to use to improve your model.
I think that's really interesting in terms of, as you had mentioned, compression algorithms. There's data that you don't need to call back if you don't need to, and making this argument that nature evolved this incredibly complex circuit, but a very efficient circuit is a really interesting idea. I wonder if this error passing is something that came naturally to like computer scientists when they did their, working on their algorithms. I wouldn't know. But to also to address some of the comments that are propping up in the comment section, whether or not we are, I think, okay, so there are a couple of comments in the comment section that are talking about whether or not we can compare neurons to logic gates. And so I'll let you give your opinion, but I want to first say mine because I am a more naive scientist. So it's it's going to be interesting to kind of contrast what I have to say to what you have to say as someone who is a lot more acquainted with the brain as an organ. I think you can simplify neurons to logic gates. It helps if you want to think of it in that way. But because biology is so complex, I think there is a danger that you over assume ways that neurons can work. As we're about to show, I think in my part of the presentation, biology always has secrets, and so you might risk acting like a certain biological engineer who believes that they can hack out their own lactose intolerance if you reduce biology to things like lines of code. So what do you think, Dr. Wendell?
Yeah, no, Fi, I think you're right. I think the best maybe analogy for this is, so I think neurons are in a way, and the networks that they work in do look a lot like logic gates. But here's the thing. So to, I guess, use another technology analogy, our understanding, I think, of where the brain is right now is that if you had let's say a circuit board in front of you, and you knew what a capacitor was, and you knew what a processor was, and you could like pick those out, and like you had a vague idea of what they did. But then you look at the rest of, your circuit board, and you see all of these things, that like, maybe that one looks like some sort of resistor, and like, I don't really know what that piece does. Like, that's kind of our understanding, is we have a basic kind of gist, but it's very superficial. It's very surface level. We're still understanding how neurons integrate all of the information that they get. As you were talking, I'm reminded of a paper that I don't even think I read, but I remember kind of reading the abstract of and being like, oh, that's fascinating, and also way above my head. is this idea that like single synapses can do like multiple complex logic operations, like within a terminal, like an axon terminal. It's not like one cell is a logic gate. It's like one piece of a cell is a complex logic gate in and of itself.
I have read that as well when I was doing neurobio. That's why it's like, I think you can, but you have to have an appropriate amount of humility, because while you can simplify things, you might need to take a step back every time you learn something new about the neuron. Aeon's got an extremely interesting comment. I don't know if you read that, or if you want me to read it out to you if you have the chat open.
Oh, I have the chat open. Yeah. All right. So how many psychoses are a result of passing forward the incorrect errors or not passing? Aeon, excellent question. We'll get to that later. Very insightful.
But yeah, I wanted to point that out because Dr. Wendell will talk about psychoses that may have roots in predictive processing. So despite being quote unquote uneducated, see, that's why I love doing these streams, right? You get these amazing thoughts coming from people that don't have the background. I really like giving you all the opportunity to flex those muscles and really push us to think deeply about some of these sciences. So great question. Very cool.
Yeah.
All right. Let's continue.
Yeah. So that's kind of my like 50,000 foot view of, maybe higher. 50,000 feet probably isn't high enough, about predictive processing. And we'll talk a little bit more about it as we get into the paper too, and hopefully flesh some of these ideas out. But I want to highlight some important and kind of cool ideas about predictive processing, as a few amongst many. All right, so the top-down models that I've been talking about represent our best guess about the cause of our incoming sensory information, right? And so predictive processing kind of posits that what we experience as consciousness or perception is at any given moment our best top-down explanation of that incoming sensory information, which I think is fascinating. There are some very interesting papers that kind of try to prove this is the case, where if you like split someone's vision down the midline and you show each one of their eyes a different picture, what happens is that you kind of go from experiencing one picture to the other picture to one picture to the other picture. And the idea there is that at each moment, you're kind of resolving one hypothesis and resolving another at the same time. And you can't quite pick one or the other because they're so disparate in terms of like what our real life experience is like. We can't see both a bowl in front of us and a barn occupying the same space. So our brain kind of has to flip back and forth between choosing which experience of reality we're going to have in any given moment.
So this is also interesting to me because I had understood it that we were sort of living in the past, quote unquote, that that there's that the lag time between sensory input and our perception is finite and non-zero. But predictive processing I don't know, and correct me if I'm wrong, but you could say that we are living in anticipation. So it's not like we're living slightly behind in like a lag, but we are creating our current present. I don't know. Does that make sense? Am I rambling? My brain is working overtime.
No. I think I understand what you mean. And so predictive processing, I think in a way can kind of bridge that gap, because you don't need to just wait for the sensory information to get there. Like you already have, the model is in your brain at any given moment, and is being kind of served up moment to moment. Yeah. I think that's cool. I understand what you're saying. Yeah.
Very neat.
It is. Very, very cool. All right. So that's one kind of idea. So this next bit is about like action, and like the role of action and how we create action. So in predictive processing, action is how we obtain new sensory information to confirm or to challenge our dominant hypothesis, right? So instead of our experience of the world being driven by the actions that we take, the actions that we take are driven by our understanding of the world in the moment, and the actions we perform are all in the service of resolving some uncertainty that we have about our environment, about sharpening the incoming data that we're getting, picking a new stream that we think would be the most reliable, essentially, the most accurate, the most precise. And this action then is driven by whatever our dominant hypothesis is. And this kind of foreshadows a little bit of the psychiatric aspects of this, where if you have some really kind of odd, wacky, top-down models about how the world works, what your place in it is, dangers, suspicions, whatever, or even ideas about how other people treat you, or how threatening or how safe you think you can feel in a certain situation, I think you can start to see maybe how distorted versions of these models could manifest as mental illness. But more on that later.
Yeah, I can't wait to get to that conversation because I find that super interesting.
Yeah. So this next bit, which will be very relevant to the paper that we talk about, is this idea of concentration or attention, right? Which has always fascinated me, is like, what is our internal ability to kind of direct our either our thoughts or our focus on particular aspects of our experience. I have ADHD, so this is like constantly on my mind. What's wrong with my brain? Why is it broken in this particular way? So predictive processing also has an answer for this. It says that concentration or attention is how we turn up or down the volume, the gain on error signals from particular pathways. This is really cool because if you can imagine a system in which you can really dampen all of this incoming sensory information that you're getting, it might allow you to do things like introspect or dream or imagine or contemplate how others will behave or are likely to behave without acting, without doing the thing. You can kind of simulate your expected sensory experiences and without having to actually have that sense data come in and disrupt your current top-down hypothesis.
I really like this top-down idea, and I can't wait to see what you have to say about hallucinations, but we'll again get to that later. I'm just really excited.
Yeah. Me too. So all right, and then so this is the tail end of this, and something I want to drive home because people, if you're anything like me, especially before I really got into psychiatry and started to have a real deep appreciation for, the things that we or I might have called before kind of irrational, emotional sorts of thinking, intuition, that all of that unmeasurable stuff that we as scientists really don't like because they make our lives so much harder, and they make doing science even worse, especially science involved with human beings, dear Lord. But I want to hammer this idea home. So top-down models can be so powerful that they can prevent us from perceiving reality as it actually is, even when we know what it is, even if you can explain it to yourself verbally and logically, right? So there's no better way to show this than with an optical illusion. And this is a really simple one here, right? So if you look at this image and you look at these squares labeled A and B, your brain is telling you, unless you're a very strange individual, in which case I'd love to talk to you. Someone would love to do a study on you, I'm sure. A and B are the same color. They are exactly the same color. And I can tell you that, and you can look at it, and you can be like, Nils, what are you talking about? They're obviously not the same color. A is clearly much darker than B is. What do you mean? And then I can show you this picture of A and B. And I can show you that this gradient is the same, right? Now, I can't do this, I don't think, very well on my screen, but if you guys can take your fingers and put them over the bar that's connecting those two checks, you will then again perceive them as two different colors. And then you take your fingers away and, oh, they actually look like similar colors. And I can put them next to one another on a slide, and you can see again that, like, you can look at this image on the right, and then you can look at this image on the left, and you can't convince yourself that they're the same color. You can't convince your brain to perceive them in the same way. And why? Like, why is this the case? Why are our brains so dumb like this? It's because our brain is optimized to predict accurately. It's not optimized to experience accurately, right? In almost every other situation in our lives, outside of this one weird optical illusion, it makes perfect sense for us to look at these lighting conditions and say, okay, things have a certain kind of reflectivity. They behave a certain way when they're in shadow versus whether they're in brightness. Like, I should perceive these two different squares as different colors. And that works in every other situation except for this one. And so you can tell your brain not to perceive it this way, but your brain is like, dude, no, I'm not going to like unlearn this very important set of rules about how your vision is supposed to work for this one dumb trick. Do you know how much of a pain in the ass it would be to try and like rework all of these neurological circuits? It's essentially saying, I'm here to predict properly. I'm not here to experience as accurately as possible. And I think this challenges our current understanding of a lot of different brain area functions, because we just assume that like there are parts of the brain that like, oh, this one's for recognizing faces, and like this part of the brain's for reward. And I think this whole idea about it, the brain is there to predict properly, is really upends a lot of this kind of sterile, mechanical sort of, you know, this is what this part of the brain does, that's what that part of the brain does schema that we have right now.
So, Thomas, the thermonuclear bomb has an interesting question, perhaps application of the predictive processing model. I don't really know anything about blindness, but Thomas is asking whether or not some people who are going blind maybe are not aware of them going blind until they are explicitly told. So I'm wondering if predictive processing has a role in maybe extending the perception of vision.
Yeah, well, so I'd have to think about the answer to that question. The broad answer is that if you accept predictive processing, or you think it might be a useful model of thinking about the brain, then predictive processing has a role pretty much everywhere in the brain, because it's a way of explaining how the brain works writ large, not any one particular sensory modality. So all that to say, it's an interesting question. I'm sure that it does have something to do with how, where our attention is in terms of the visual information that we get. It's often not from our periphery. And so you're not likely to be refocusing your attention in a way to kind of adjust that sort of prior that you have about your peripheral vision being fairly normal. But I'd have to think about that more to give a good, not hand-wavy answer. Yeah,
and I also wonder if that counts in your field, or if that's more neurology.
I mean, yeah, that certainly does lean more towards the neurology side of things.
I just say that because I've had for a while a difficult time understanding the difference between psychiatry and neurology. So I think, you know, as a complete naive person, you know, just like, y'all are brain people.
Oh yeah, no, that's all right. At one point when I was in medical school, one of the neurologists was lecturing us, and he was like, asking the crowd for questions, and I raised my hand and I said, how long do you think before neurology and psychiatry are the same field? And he just kind of laughed at me and he said, oh, that'll never happen. And I'm sitting here thinking, like, it's the same organ, my guy. Like, what are you talking about?
I think that's really interesting too. Just the brain is so hard that we needed two specialties.
Yeah, right. Exactly.
Very cool. Very cool. All right.
So yeah, that's my spiel on
Great.
this.
Sounds good. Let me go ahead and get the next presentation up. So you don't have to have understood every bit of that to understand where we are going to head in our discussion, but that was a really helpful primer in our to kind of appreciate where we're about to go with all of this. So I'm going to throw up my presentation now. Now we're actually going to be talking about the paper. And so I think a commenter had asked, how are we going to test this experimentally? I think that is something that will be difficult to do for all of predictive processing, but as we do in science, we can pick at the problem little bit by little bit. And so this is one of those papers. So I've actually had a bit of difficulty on agreeing which paper to do with Dr. Wendell here, just because a lot of the predictive processing work has been, and you computer science nerds in the chat will love this, they've been theoretical computational studies. But I'm a molecules kind of guy, right? I wanted to pick a paper that was in line with the stuff that I had been doing on my channel, namely thinking about the world of molecules. And so this is a real experimental biology paper that investigates a little bit. Not exactly. I wouldn't go so far as to say that this rigorously tests hypotheses of predictive processing, but can help us understand how predictive processing might work. And we can get into sort of the nuance of that truth as we get into the paper. So on the screen here, I have my self-perception of brain size. And let me tell you, I have a background in neuroimmunology, but So that's why I felt comfortable with agreeing to do this paper, because I was like, oh, I'm a neuro person. Surely, like, this neuroscience paper is going to be like no problem, right? In reality, this was the hardest thing that I've read maybe in half a decade, maybe ever. In at least half a decade, this is the hardest paper I've ever read, because I came to the crushing realization, and I've been going back and forth with Dr. Wendell over Discord begging for help, because I realized that my entire neuroimmunology background was basically treating the neuron as a fancy cell that shoots electricity. That's it. I didn't have to think about circuits or systems or perception or lived experiences. My neuro background is the cell, the neuron, it goes zap, zap, and there are immune proteins sometimes. That was the extent of how deep I was going. So this was my first real foray into like thinking about the brain as a functional organ and not just a mass of funny cells that act funny. So that is going to be my primer. Now, I've worked quite hard, and I think Dr. Wendell can attest to this, having been on the receiving end of many of my messages, to make this as understandable as possible. And I think I've done a pretty good job at it. So don't worry. I've done my best to try to teach this to myself and to have Dr. Wendell teach me, so that we can get into understanding the world's most complicated organ just a little bit more. Alrighty. So let's get into this. So the neurotransmitter dopamine is responsible for all sorts of different things in the brain. And the reason why I chose this paper is because it had dopamine in the title. I was like, dopamine, that's a molecule. So that means that informed me that this was not a purely computational paper, and that would actually be able to visualize cells and stuff. So we're going to be investigating the role of dopamine in this predictive processing framework. All right, so it's responsible for, or it aids in things like movement, reward, and learning. And what this paper is going to do is it's going to refine the role of dopamine in a very specific type of learning that is going to be modeled in a predictive processing background. To understand this predictive processing, we have to first talk a little bit about prediction error. And again, this is something that I think a lot of you computer nerds will get hyped over. So as Dr. Wendell had said, predictive processing is about managing errors rather than reporting reality. It's just a more efficient system. And so we can start to think about what kinds of prediction errors the brain can receive. And one of these prediction errors is called a signed prediction error. And so signed prediction errors, and of course Dr. Wendell will correct me anytime that I'm wrong, signed prediction errors have a sort of what stakes to them, maybe. So some sort of positive or negative reward for the subject in question. So if I'm playing Genshin Impact and I'm expecting to pull the 5-star banner pity, and what I want to pull is an interesting, good 5-star character, and I get Hu Tao, I'm happy, I'm pogging. That's a positively signed prediction error. So this is associated with a measure of reward. And so in classical cognitive models, this is associated with an increase in dopaminergic firing. So neurons that produce dopamine start dumping their dopamine out. That's causing neurons to fire with one another, causing those neurons to strengthen those relationships, to strengthen their firing, and therefore causing learning to happen, right? So this is, I'm not saying this is how this works. I'm saying that this is actually how I learned it back in graduate school. So this is sort of the classical way to understand this. Fi is addicted to gambling. So I actually am not, because as we're about to see, there's also the opposite side of this coin. Negatively signed prediction error decreases dopaminergic firing. So again, I'm playing Genshin Impact. I'm expecting to pity on this banner, but instead of getting Hu Tao, I get Chi Chi. So this is a real story that happened to me while I was playing Genshin Impact. And after I lost the 50-50, I quit Genshin entirely. So I am not a gambling addict. When I am punished, I do not go back in for seconds. No, thank you. So in this model, if you have something that is negatively signed, so something that results in sort of a punishment of sorts, I don't know if that's technically correct to say, but if your expectations were not met and that was bad and that sucked, then that's a negatively signed prediction error. And so that would classically result in the decrease of dopaminergic firing. So you withhold that dopamine, withhold those good feelings so that you don't learn to do that in the future. So that's how people, I believe, thought things happened. All right. So Dr. Wendell was careful to point out that sometimes some neurons actually increase their firing when you have negatively signed prediction errors as well. So it's not clean. Why would it be clean? It's the brain. It's the messiest organ to ever exist. But just so you know, the brain is very complicated. Even like models like this cannot capture all the complexity. All right. But there are more than just things that result in correct predictions that are good or predictions that are bad. There's also completely neutral predictions, right? So if you have a situation where you've seen your friend Joe wear red for the last 3 Tuesdays, today is Tuesday, and you predict that Joe will wear red, but he shows up wearing blue, unless Joe has made wearing red a core component of his identity, you're probably not going to have a positive or negative reaction either way, right? So this is what's called an unsigned prediction error, one without an obvious reward of whether or not you would predict it correct or not. I say that very carefully. Obvious, right? Because you can argue that making a prediction correctly is rewarding on its own, but for the purposes of my talk, I'm not going to get into that, although maybe Dr. Wendell can elaborate later. Okay. So classic cognitive models, the way that I learned and the way that many people have learned and the way that is easiest to think would suggest that dopamine would not have a role in unsigned prediction errors because there's no inherent reward, right? Like guessing that Joe is wearing a red shirt correctly doesn't actually benefit you in any kind of way except just by virtue of being right. So this can be thought of as just kind of surprising.
Yeah, and if I can add a little color, fine.
Yeah, go ahead.
So I think another kind of important concept here with this surprise is that part of, in the classical model of when you would see dopamine firing or not, is all relative to like what your expectation was in the first place. So like you said before, if you had one expectation and actually exceeded your expectation, dopamine would be released. Here, with like surprising stimuli, you don't actually have an expectation in the first place with certain surprising stimuli. You don't even know that it's going to happen. So you have no way to identify like what the baseline expectation would be. It would be like somebody new walks into the office even, and you see that they have a blue shirt on, and it's, if you see dopamine firing there, then the question is, well, why? You didn't even know the new person was going to walk into the office.
Yeah, that's also true, and that'll actually apply a lot more when we get into the actual experiment here. Um, so in this model of predictive processing, we actually find that, or rather, experimental results will show that these unsigned prediction errors, these prediction errors that are not associated with a benefit or a loss, also evoke dopamine firing, which means that dopamine, despite being the molecule that we think of as the reward thing, is actually playing some kind of role in these neutral, quote unquote, responses such as surprise. So I want to give Dr. Wendell a little time to talk about this ongoing war between the concept of signed versus unsigned prediction error, because from what I understand, you might not need to explain away learning on the level of reward, or so I think you can do a much better job of explaining this than I can. So why don't I give you the floor here?
Yeah, I will do my best. So the idea with predictive processing is that we don't necessarily need positive and negative salience attached to error prediction. We can just say that they're unsigned errors, and what's important is the magnitude of the change that we didn't expect, right? Like your top-down models are kind of agnostic in terms of what the flavor of the error is that shows up. I'm not sure. Yeah, I think that's basically the idea, is that you don't need emotional salience attached to it, or there's this idea that like the emotional salience is kind of what makes the rewards, kind of stick or makes you learn from them a little bit more. Predictive processing maybe has some things to say about that too, which I don't have enough of a grasp on to fully get into. But suffice to say, predictive processing just says, we don't need signed prediction errors. We just need prediction errors full stop. And we can do all of the things that predictive processing needs to do with that information without emotional salience attached to it.
That's not to say that emotional salience doesn't have a role in predictive processing, right? So I just want to make sure that that language is clear.
Correct.
Right,
yeah. And there are interesting ideas about what role emotions play. Some people think that, emotions are a way of directing attention in certain situations, but like both on a positive and a negative sort of scale too, right? If you're joyful, you have your your feelings of anxiety, uncertainty, all of that. But the role that they play is thought to be a little bit different. They're more of a consequence of action and of rising error signals than the cause of, per se.
Yeah, Aeon's got an interesting hypothesis about dopamine's role in these surprise learning, in evolutionary role, making sure that when something expected happens, that you are rewarded maybe for a correct prediction when the threat is low, kind of like training wheels, which I think is interesting. Don't know the answer, but that's an interesting thought.
That was actually, so in this kind of more classic model, that was part of the explanation for why dopamine was released during surprise, is that there is something that should be inherently rewarding about experiencing surprising situations because it helps to drive learning.
Very cool, very cool. All right, so let's move on. Okay, so this is an important part of science. Just want to hammer this. There's something that my advisor used to tell me all the time, and that's all models are wrong, meaning that reality is infinitely complex, and in order to understand reality, we have to the limits of our brain makes it so that we have to simplify reality. So in that way, every model that you come across is wrong in some kind of way, but that doesn't mean that they're not useful. So don't be a weird anti-science person, right? People complain that climate models are wrong all the time. Well, models by definition are wrong when you're faced with objective reality, but that doesn't mean that they can't be used to generate some accurate predictions, right? So keep that in mind, that just because that old model of dopamine and signed prediction errors, just because that might not reflect reality in its entirety, doesn't mean that it can't be used to make accurate predictions either. So that's just a meta science kind of idea. Okay. So we talked about these signed and unsigned prediction errors. Experimental results indicate that surprises, which have like supposedly no emotional salience to them, also induce dopamine, as we had said. So I like this meme. So when you get your expected stimulus, you're a genius. You get that dopamine hit that's rewarding you for making a correct prediction, but you also get a dopamine hit when you are faced with unexpected stimulus. So the question is, well, if dopamine is released in response to both types of responses, well, then what the heck is dopamine actually doing to begin with, right? It's not useful if dopamine is always firing, so dopamine must have a sort of interesting, complex, nuanced role, and that's the kind of thing that we are going to be experimentally investigating in our paper today, which is very exciting because whenever you do experimental stuff in the brain, to me, that's more compelling than just reading code. Sorry to all of you computer nerds out there. Okay, so predictive processing has room for a role of dopamine in surprise learning. So we saw this in the introductory presentation, that we have this bottom-up sensory input flows to the brain. And so what is being passed in this flow of information is that error, and then from top down, the brain is doing, although maybe this is a little inaccurate now that I've actually seen your presentation, but basically there's this loop of reporting errors and then making predictions. And these things run until the error is minimized. So that is how we are able to adjust to our changing reality. Something happens equals dopamine. Yeah. I mean, we could end there and we would be correct, but we want to go a bit deeper. So dopamine's doing something, and that's what we want to find out. Okay, so how do we study surprise? So this is a question that I want to pass to Chad. I'm going to let you hold the reins of the experimentalist. And so say you have a neurologically complex model organism like a mouse. How would you create surprise for the mouse in a way that is experimentally useful? Give you all a couple of seconds to think about that. How would you surprise a mouse and have it be experimentally useful? That's not a trivial question to ask, because I'm sure it's easy to surprise a mouse, but in order to surprise a mouse in a way that is going to generate data that can be interpreted, that might be a little harder. Yeah, think on that for a second. Give the mouse's coworker a blue shirt. Now that would be surprising.
I think the shirt could be any color, honestly.
Yeah, just the fact that he's wearing a shirt, that would be a surprise. Food or not food? That's also interesting, but I don't know whether or not you can separate that from reward, right? Because we know that eating food is inherently rewarding. You're going to get dopamine from the act of eating. That might have a confounding variable with just the element of surprise. FNAF jump scare. I mean, that would also be quite surprising, and that's not tremendously far off from what's happening. Expose it to a predator. Also interesting, although that might involve more evolutionary survival pathways that might make the interpretation of data a bit messy. Bad tasting food. Ooh. Again, with the food, I'm not entirely sure if that is... if that would be clean enough for our data. I mean, loud noises are surprising, but also might be somewhat unethical due to the nature of animal abuse. Okay, Aeon, we're getting closer. We're getting closer to the correct answer. Actual serious answer. A tile or something that can be removed to reveal a different color. That's not a bad suggestion. However, mice don't have great color vision. Yeah. Stimulus with reward versus stimulus separated from reward. Mr. Fox, that is... This is sort of like how I classically think of it too, but we're actually trying to uncouple reward from learning. Surprises in our unsigned prediction errors have no emotional salience. So how would you surprise a mouse but not have it be emotional? And I'll give you a hint. Aeon is hovering around the correct answer. Confuse them with smells. Okay, that's an interesting idea. I think smells access a very deep part of the brain that might be too wired in survival to get something that is this clean. Let's think about sounds. what might be a way to elicit surprise with sounds other than something loud? We're hovering there. Very interesting ideas, chat. How would you, with the power of sound, generate a surprise? And I want to challenge you all to think as cleanly as possible. So things that are social, like the sound of another mouse, that is going to be wrapped up with things like evolution, sociability, all these other complex brain pathways that we don't really want involved. All right, this is good. This is good that no one is getting the answer, because the actual answer is going to be that much cooler. So I'm going to move to the next slide here. So sound is correct. Loud sound, because that can be shocking, because that can elicit a level of fight or flight, is probably a little messy. Probably can't uncouple that from reward or punishment. So here we have the experiment. This is the sound that will be played. So in this experiment, we have what are called standard tones. So we have the same note being played over and over again. The mouse has time to adjust. The mouse can make predictions on what tone will come next, because it's all of the same, they're all the same note. Now, to elicit surprise, we can throw in a deviant tone, something that is a lot different than the standard tone. So we can appreciate here how this is surprising, but it doesn't threaten the mouse in any kind of way. This is just something that, like the blue-red shirt, doesn't really matter to the mouse other than making the prediction correctly. All right, so does that make sense, chat? This is how we are going to create surprise for the mice. So over time, neuronal responses to standard tones can get suppressed. So if you hear the same tone over and over again, your neurons get kind of, and correct me if I'm wrong or oversimplifying, but your neurons can get kind of bored. So previous explanation for this boredom is that their synapses just exhaust, like they don't have any more neurotransmitter to fire at a high enough level, so they just kind of get bored and tired. But I believe in the mode that we're thinking of predictive processing is that the top-down brain is deciding not to pay maybe as much attention, quote unquote, in response to these noises. Yeah, the brain will tune out the sound. That's kind of how I'm understanding it. Although I'm like walking on glass here because the brain is so complicated.
I think a good way to maybe think about it is that the brain's, the top-down model at this point is like, oh yeah, like I predicted this already. So the top-down model, right? The prediction engine is sending signals that are canceling out the upflow of the error of what would be the error in this situation. Is it's accounted for in the model.
Yeah.
In the top-down model.
And then when you get to that deviant tone, all of a sudden, neurons are excited again, so that increases the synaptic response. And this is representative of updating your prior beliefs. So when you're hit with that surprise, then you can maybe, again, walking on glass here, update prior beliefs, I'm equating to rewiring, although, or readjustment of firing rate or neuronal activity, maybe broadly. I'm trying to be as broad as possible to not be wrong.
Yeah, I think that's probably pretty fair. I would just highlight in terms of like the learning just for chat as they go through this is often there's this tendency to think of it as like purely rewiring and very kind of complicated, more sort of permanent long-term stuff. Neurons have the ability to actually make pretty subtle changes that don't require rewiring on very, very short time scales that might just last for a few minutes or a few hours even. So the learning can be very in time terms pretty short, but it's still learning in a sense.
So Thomas is an interesting question. Maybe it's just a personal or human thing, but I would be kind of bothered if there was a repeated note and then it randomly changed all of a sudden. I don't really know if so this is new to me, but given that this is a model that exists already, I'm sure that they have everything optimized so that we are not bothering the mice with too long of a training period, is my guess. But that is an interesting experimental variable to consider, whether or not we are instead eliciting some kind of frustration response or something. Worth keeping in mind. Okay, so next slide. So that's how we're doing surprise. So again, we're asking the question, what the heck does dopamine do as we are learning to deal with that surprise? Or again, quote unquote learning, how our neurons, I should say how we are reacting to surprise neuronally. That's maybe more accurate. So up until here, I wrote down my naive hypothesis. So at this point, I didn't finish the paper. In fact, judging by my conversations with Dr. Wendell, I hadn't even understood the paper at this point. So this is my hypothesis. Feel free to come up with your own. But my guess was, well, I know that dopamine plays a role in learning and memory. That's like one of the first things that we learn about dopamine is that it well, there's the reward thing, and maybe the second thing is that it helps you learn. And so my naive thinking here, or my naive guess, is that maybe dopamine is released when the brain believes we need to learn. Like when something changes, that's when we need to learn. Like maybe there's this like, okay, if everything is the same, then we don't need to learn as much. And then when something different happens, okay, we need to learn. Let's turn the learning machine on. And we turn the learning machine on with dopamine. So it can be in the form of, we're right, let's strengthen the connections that are associated with our prediction. Or if we're wrong, let's redo some of what we've learned before. So maybe dopamine is some sort of molecular enabler of change and not necessarily a reward, as we have been talking about separating this idea from reward and learning. All right, so this experiment goes a little further than just this surprise tone, which is called the auditory oddball. So the playing the same tone and then changing the tone, that's the auditory oddball. To sort of uncover what dopamine is doing during all of this, we are going to inject dopamine into a circuit of the brain. So putting dopamine in the area of the neurons that are going to be responding to this change in sound. So, you know, we have this tone that plays, and then we get that surprising tone, and then we inject little bits of dopamine in the brain, and then we will record the response of those neurons to those surprising sounds. Okay. So here is the first paper, and so this data represents the response of dopamine to sound. So this is not the auditory oddball. This is just, we hooked a mice up to some brain recording technology, and we played different sound tones at different volumes as well as different pitches. So on the y-axis, you see that we have intensity, so that's decibels, so that's how loud the sound is, and then frequency, so that's how high-pitched it is. The higher frequency, the higher pitched it is. And so the color, the heat map that you can see on the right, the more red it gets, the more activity we see. So in this graph, we see that there are different populations where there are neurons that after you've injected dopamine are either more active than before or less active than before. So this is one of those pieces of data that makes me go, oh God, the brain is so complicated, because, you know, there's not a unidirectional effect of dopamine here. Some get more active and some get less active, and this to me was already very confusing because if you come at it from that classical model, right? Like dopamine increases firing, you learn; less dopamine, you learn less. This already flies in the face of that idea, which is why I think I struggled so much with this paper, because I had to unlearn a lot thinking about neurons in this context. Pretty interesting. Okay, so I believe they use this as a way to set up their experiment down the line as well, although I could be wrong on that.
Yeah, no, I was actually thinking through this earlier today, Fi. So my impression is this is just them showing that like dopamine has the capacity to change the response of these cells to sound, because if you can't show that, then like what are you doing?
Yeah, a good first experiment. This data set is all over the place in my eyes. Yeah, the more you do science, the more you kind of get used to figuring out new data, but this one was kind of confusing. You're looking at the color, and then that'll give you an idea of how much activity there is at that neuron. So I'm an angry neuroimmunologist because this is how they presented their data. And I want to give chat a little bit of time to think about how a neuroimmunologist might react to seeing data that is labeled this way. So it's not that I have an issue exactly with the... Dr. Wendell, I think we can hear your keyboard clacking.
Oh, sorry.
Yeah, no worries, no worries. So I'm an angry neuroimmunologist because this is how the data were presented. So if we are hooking a mouse to what is essentially a neuron reader, and we're saying that there's this control and then you've injected dopamine, does anybody in chat have an idea of why I'm frustrated immediately by this graph? And previous stream watchers, I think, might have an idea of why this graph kind of pushes my buttons. Let me put it this way. Is there any information here that is missing, or you would like elaborated, chat? What are we missing here? X-axis is not linear and is not logarithmic. I'm willing to forgive that, because if the data is not readable by eye in an axis that makes sense, you can sometimes take liberties. For example, if there was more interesting variation between 1 and 10 hertz, then you want to show that. And then if there's not as much interesting variation between 10 and 42, then you might not need to show that. At the end, science is still about communication. So that's not the axes. What's a basic question about this experiment that I have not explained? And again, if nobody gets it, that means it's a good question. That means you're going to learn what my pet peeves are in terms of science. So that's not a lost cause either. All right, couple more seconds. What's missing here? Chat, does anybody know what the control is? What's the control here? They label their data with control, but what is control? And I'm not accusing the authors of not explaining their control. They do, but on this graph, they don't explain what the control is. You don't know, right? It's not immediately obvious. I have been begging my students to develop a habit where they label their control, because the word control is useless. Control is the group where nothing is experimented on. You would think that. You would think that, Aeon, control, that's the definition of a control, but actually controls can come in all sorts of different shapes and sizes, which is especially relevant for someone with neuroimmunology background like I do, because there's something that a lot of neuroscientists take for granted about doing things like hooking up a mouse's brain to wires and stuff. That's, if you are reading from neurons, you've pierced the skull. You know, you have changed the immunological environment of the brain just by taking your measurement. What does dopamine control mean? Exactly. Still trying to read this graph. Don't worry about it. Just the redder, the more activity, the bluer, the less activity. But yeah, A's got it right. What does dopamine control mean? It's not explained in the graph itself, although it is explained in the paper. Just as future scientists, make sure to label the control in the graph. Just makes it easier to read. So what the heck is this, right? What is the control condition that we are comparing dopamine to? So in their method section, they have here the dopamine manipulation procedure. So the control condition is what happens before any dopaminergic manipulation is introduced. So up until the point where dopamine is injected, that is what the control is, meaning that the mouse's brain has already been sort of penetrated by instruments. The mouse's immunological environment is changed. So that's not comparing a mouse with dopamine to a mouse without dopamine. It's comparing more carefully a mice that has had its brain wired to a mice that has had its brain wired plus dopamine. And that might be like an annoying bit of, information, but when you're a scientist, this kind of detail is very important, so make sure you always label your controls. Controls are very important, and choice of control actually matters. So here I am going to do a little bit of a poll for the audience here. I have 3 examples of what could be controls. And this is an illustrative example of why thinking about the appropriate control is a worthwhile scientific endeavor. So we have the before injection condition, like we do in this paper. So you hook up the mouse and then you do some readings, and then you inject the dopamine and you do some more readings. And then the second control, number 2, sham surgery, basically means you do the surgery and then you take your readings and then you end, which for this case is the same, or vehicle injection. So instead of injecting dopamine, you inject something that is isotonic to the brain environment, meaning that it won't change the brain chemistry in any way. Like you inject dopamine, and then your control is injecting salt water, like a normal kind of fluid that the brain might have, cerebrospinal fluid, something like that. And then no treatment, so not doing anything to the mouse at all. So with that in mind, chat, I'll put up a poll, and I am going to ask you, which of these options would you pick as the appropriate control? So 1 is original, and then 2 and 3 are the ones that I have put in as ideas. So before and after injection of dopamine, that's one control. Injection of salt water, that's another control. And then no treatment at all is another potential control. And before I get into what I think, I want to hear from chat if you want to offer. Oh, a bunch of you have already voted. If you have thoughts as to why you have chosen what you have chosen. And then I'll turn it to Dr. Wendell after chat has had their piece. So if you reason out why you chose your control, even if you're right or wrong, if you verbalize why you have chosen this specific approach, if you're right, I mean, we're talking about learning, right? If you're right, if your expectations were matched, you'll increase that synaptic strength, you'll learn. But if you're wrong, you might learn anyway. Okay, so we have now a little bit of disagreement. Most of the people in chat are choosing 2. Okay. So that is very interesting to me. Dr. Wendell, do you have thoughts on which of these controls is the appropriate one to do?
I mean, I haven't had to think about doing this kind of stuff for a while, so my inclination is to defer to whatever the authors chose to do in terms of their control, to be perfectly honest.
Mm-hmm. But I'm challenging you because I have been challenged by your choice in paper.
I suppose that's fair enough. I mean, I think the before injection, in terms of control, makes a good amount of sense, because in that case you are measuring the exact same neurons. When you do this, right? That's how their experimental model was put together. It's literally the neuron before you inject the dopamine, the neuron after you inject the dopamine. You don't get realistically a better control setup than that. Whereas, like with sham surgery, vehicle injection, I mean, you'd have to really kind of change up how you were doing the experiment. And practically, it might be quite difficult if you have to wait for washout periods on the same animals.
Yeah, so let's go ahead and see what my thoughts are. Not to say that I'm correct. So there are some pros and cons of each process. So before injection and after, as Dr. Wendell had pointed out, you can compare within the same mouse. You can compare within the same exact neuron. But as a con, you cannot rule out individual correlates. So you are comparing within an animal, which can be quite not satisfying, because you are treating basically neurons as your ends instead of animals, which I think in a lot of what I do, I would prefer normally, right? Because on an animal to animal basis, there are so many covariates between individuals that you might over exaggerate differences and find effects that might not actually realistically be there. For option 2, which is the most popular option by chat, and I'll actually now end the poll there, so you've all locked in your answers. The pro of sham surgery / vehicle injection is you can control for neurotrauma and inflammation. The con is you can't get as much precision because you have to do so many more mice, because it's unlikely that you'll be able to compare apples to oranges, apples being mouse A and oranges being mouse B, right? But by injecting the same volume, if you're injecting volume into the brain area, you are pushing the neurons around. You are effectively kind of hurting them. And so to make sure that whatever weird brain effects isn't caused by an inflammation response to the bit of liquid that you injected is something that I care a lot about. I'm glad that nobody picked no treatment. No treatment would be a terrible control. The pro, I guess, would be for animal safety. I mean, you didn't have to do anything to a mouse, so that's kind of cool. But this only has power if the sham vehicle approach is also done, so it could potentially explode the number of animals that you would actually need to use. It might surprise you that as a neuroinflammation person, I actually pick... Oh, it didn't show. Okay, so yeah, okay, never mind. There's a bit of discussion before I reveal. So how important is neuroinflammation in this context? So research has shown that immune molecules in the brain can affect synaptic plasticity and neuronal communication. So not a lot of people know this, but in the rest of the body, the immune system does immune system things. It helps us fight bacteria, viruses, fungi, pathogens, cancer. That's what the immune system does everywhere else except the brain. The brain is a very, very special place for the immune system. And the immune molecules, while they might be fighting infection in other parts of the body, is actually helping your cognition in the brain. So the same molecules that my audience has probably heard of the attack complex, right? Those molecules of the attack complex actually do different things in the brain, which is really interesting. The complement proteins, right? Complement proteins in the brain do things like prune synapses on microglia. So it actually, not on microglia, I believe they prune synapses on neurons as well. So in the learning process, as neurons are removing certain connections, they actually use some of those immune system proteins. So of course, I would care a lot if the immune area or the immune environment of the mouse has changed. So this could potentially dampen or exaggerate effects, right? So we don't know. I don't have a particular hypothesis, but if you induce inflammation, you could potentially change how strong these effects can go. So does this ruin this experiment? Chat, I want to give you another chance to think about whether or not neuroinflammation is useful to think about in this situation. So think about the way that the experiment is set up. We have a control condition where you do everything except inject dopamine, and then you have the after condition after you've injected that dopamine. So, is this bad experimental design? Is neuroinflammation going to affect this particular system? Is this deadly? Can we trust anything that comes after this slide? This one's a much harder question, I think, to answer. Is this deadly for this experiment? I take back calling chat the compliment system. You should call it the insult system. Oh, I like that. I like that a lot. Oh, I love calling chat the compliment system. I don't want to call them the insult system because they're gonna insult me. I would think, yes, as moving the neurons may be damaging. Yeah, if you inject that little bit of volume, you could be moving things that could be damaging. Could be. It's definitely true. Inflammation tends to have an inherently negative connotation. In the immune system, in the brain at least, you need those immune proteins for your neurons to fire correctly. Very, very interesting. Yeah, so...
Well,
what's up?
Sorry, I was going to say, while chat's thinking about that, I was just going to add 5. There's actually fascinating that you should mention the role of immune proteins, because there's a whole literature on schizophrenia. And if you go and you do these big like GWAS, genome-wide association studies, a lot of the SNPs that are abnormal and individuals with schizophrenia are all like complement proteins,
Yeah.
MHC proteins, that sort of
Yeah.
stuff. So,
And
yeah.
that was actually a lot of the focus of my experience in graduate school, is thinking about ways that the immune system and the brain function with regards to schizophrenia, brain injury, that kind of stuff. I love that level of detail. Not that I love that schizophrenia exists, right? But the fact that schizophrenia is such a big like molecular mystery, the fact that these proteins in the brain that nobody even thought existed inside the brain for a long time is the strongest, or one of the strongest, molecular correlates of developing schizophrenia is really, really interesting, and might be something that I do for a journal club one day. Okay. I'm saying no because there is another slide. Thomas has been one of my audience members long enough to know when I'm about to throw a curveball. I think the immune system could completely change the outcome. All very fair. And I love this split because science is very nuanced, right? Like, there are arguments to be made for either case. And sometimes it takes experience to know which is the correct answer. But I think I'll cut the poll here. We're about split, with the majority of chat saying that this is deadly for this experiment, that we can't really trust what comes after here. Okay, so it might be surprising for you to learn that as much as I harped on this neuroinflammation thing, I actually still agree with the original authors, right? I still like this pick because the power of recording the same exact neuron before and after is too good. It's too good to really be that concerned about neuroinflammation. That's not to say that you shouldn't be concerned with neuroinflammation, but think about it, chat. The only difference between the control group here is whether or not you've injected a very small volume of dopamine. And yes, that little bit of volume could be damaging, and it would not be a bad idea to do another set of experiments where you only inject saline. I don't think that's enough to outweigh the potential power of this experiment. So as a neuroinflammation person, am I a little annoyed that there wasn't a vehicle injection? Yeah, I'm a little annoyed, but I think we can get away with it because they did the entire surgery too. So the surgery is the same for before and after, right? Because it's the same mouse, it's the same neurons. So the major inflammation is the same. It's just whether or not injecting that bit of dopamine causes additional inflammation. And I think in the grand scheme of things, injecting a tiny bit of volume, that might not be as crazy as opening up the cranium and tearing back the skull or whatever they had to do for this experiment. So while I think you are right, the immune system could completely change the outcome. It is a legitimate fear, but I think the way that this experiment is set up, we don't really have to worry about that too much. Tiny volume equals tiny inflammation. Yeah, or at least that's what I'm hoping, right? Yeah, they should have, but if you think about the value of animal lives, is it a big enough deal to do another set of mice and sacrifice their lives? It's arguable, right? In research, we want to minimize the number of mice that we use. We don't just use mice all willy-nilly because some neuroinflammation person got mad. So I do see, but I want to chat to think about the design of controls because I think that's a very useful experiment, thought experiment to do. Okay, so with that detour, let's continue. So what do the dopamine supplemented neurons look like? So here we have our surprise, our auditory oddball experiment. You play the same tone for the mice, and then you hit them with that surprise. And so we have on the bottom axis a measure of the ratio between the pre-surprise and post-surprise responses from the neurons for the neurons before they were injected and then after they were injected on the y-axis. And this is, and Dr. Wendell can confirm, a figure that I had a lot of trouble with. So I think the best way to understand this is to first look down the y equals x dotted line. These neurons in gray, these neurons did not really change their response in response to the surprising tone. So these are neurons that didn't really change all that much. If you see purple dots that are very far away from this straight line, it means that they've had a response that is greatly different, whether or not they had dopamine or not, right? So there are some neurons that before you injected dopamine reacted very strongly compared to the not surprised tones. And then after dopamine, you don't seem to see quite as many of those neurons. So if I could describe it visually, it's like more of these points got dragged down to the bottom right, whereas not that many got dragged into the top left. Does that make sense? Okay, but what does that mean? So dopamine supplemented neurons get quote unquote less surprised. And so this is an oversimplification by me of the CSI index, which is the ratio of their activity before surprise and after surprise. What are the dots? It's the ratio of surprise to not surprise. So out here in the far right, these are neurons that are that are reacting very differently in response to surprise. And then as you get closer to this line, you get neurons that don't really react all that differently after surprise. If this plot is confusing to you, don't worry. There'll be a bit of an easier graph, I think, to interpret in just a little bit. But this is kind of the raw-ish data, minimal processing. Why are there two colors? Purple are neurons that reacted very differently, and the gray neurons didn't react very differently at all. Okay. So let's break down this data and take a look at the violin plots. So violin plots, as we'll see in just a little bit, allow us to get a population level understanding of the different reactions that these neurons have. So the x-axis here, this is distribution / frequency, and then the y-axis is magnitude. So the taller this graph is, the more these neurons are reacting. So in this dopamine control, so on this left-hand side, we have the control. So this is spontaneous firing rate. This is just the neurons. They're just hanging out. No surprise tone. Just quiet room, mouse hooked up to a brain reading machine. And here we have not a lot of neuronal activity, right? On the left and right parts of this graph, we get pretty much the same whether or not you injected dopamine. So when there's no tones at all, the neurons are not really that active. Okay? I hate violin plots. I did too when I first learned them. They were quite difficult to get into. Now let's look into what happens when the mice hear the deviant tone, right? So you play the same note, and then you get that surprising note. So in response to surprise, now the mice that are injected with dopamine do react differently, right? Chat, take a look. The violin part of this half has been dragged downwards, right? So we see that when you get that surprising tone, that the control mice that don't have dopamine injected, their neurons start freaking out. They start spiking like crazy. But the neurons that have had dopamine exposed to them, their neurons don't fire quite as rapidly, I should say. So that to me is quite interesting. So then you might wonder, does this change the response in response to the tones that are the standard tones? And the answer is no. So here we already have a very interesting role of dopamine in the response to surprise. So in the control condition where you're just chilling, no music is playing at all, dopamine doesn't do anything. When you play the tones that you get acclimated to, the predictable tones, dopamine doesn't do anything. But when you are surprised, dopamine actually stops your neurons overall from firing as frequently. So that is pretty interesting. And I'm going to pause here because it's pretty important for the rest of this data that we understand what this plot means. My brain is smoking. I know. Violin plots I feel like are so brain breaking. Basically, how thick the violin part is, that's the distribution. So the thicker this part is, the more neurons are responding. So if we go to the top here, this very thin neck of the violin means that there are just a few neurons that are firing at a rate of like 30 spikes per second. But here, at the thickest part of the violin, we see that most of the neurons, when in response to the shocking tone, are firing at just 5 spikes per second. You can see between the control and the dopamine that overall, the dopamine neurons are firing slower, which I think is pretty interesting, right? Okay. when we take a look at Dr. Wendell, there's some crackling on your end.
Oh, that's just me.
So when we take a look at individual neurons, we get an interesting picture of what's happening. So overall, dopamine stopping neurons from responding as rapidly or as heavily to surprising sounds. But when we take a look at neuron by neuron, we see that there are interesting distinctions. And by that, I mean incredibly hard to wrap your head around individual changes in activity. So here we have the CSI. And so don't worry exactly what this means. Just take a look at these peaks or the red and the blue peaks. So the red peak represents activity in response to the deviant tone. The blue is the standard tone. And so the ratio of the surprise to the deviant tone is the CSI. It's basically how much did the neuron change in response to surprise, right? So we can see that there are two distinct populations of neurons that respond differently to dopamine. So control, so this is before you inject dopamine, and the red and the blue peak get a lot closer to one another. So the CSI decreases. So normally the neuron would like freak out and start like firing very quickly, but dopamine actually prevents that from happening. It actually causes the ratio to decrease after hearing that surprising sound, and that normalizes to some extent after they are allowed to recover. But not only do some neurons close that bridge, there are other neurons that exacerbate that bridge, right? So this neuron on the bottom here, this neuron in the standard tone, that neuron did kind of react to the standard tone, but it also reacted to the deviant tone. And after you injected dopamine, now that standard tone doesn't get as big of a reaction, but that deviant tone still does. And so the overall result is that this neuron's difference is much larger. CSI is the difference. I think it's the ratio. Dr. Wendell can correct me if I'm wrong. I think it's the ratio.
It is. It's the ratio. Yeah.
Okay, great. Thank you. So even though overall the neurons exposed to dopamine are not firing very quickly, if you look at a neuron by neuron level, some are actually increasing their response to surprise, some are decreasing their response to surprise. And that is unexpected to me, and it breaks my brain to even think about it. So if I were to simplify it, I would say that dopamine causes a complex reaction in response to surprise. Overall, neurons are quieter in response to surprise, but there are individual differences. Anything you want to add, Dr. Wendell?
Yeah, so I was just going to add, so I think first, like you said, Phi, it's important to just see that there are differences in this change of CSI, this ratio between how much response you get to a deviant tone versus how much response you get to a standard tone. But the big take home is that doesn't matter what cell you are, your response to the deviant tone is going to decrease with dopamine administration. The other thing is, so like CSI shows up in this paper all over the place, and I tried really hard to figure out why the authors cared so much about CSI. They don't really explain it. I don't really understand why they think it's so important. I'm guessing that it encodes some sort of information that they don't really explain very well, but I think reading and thinking about this paper in the predictive processing context gets a lot easier if you don't worry about the CSI so much, because I think the take-home messages are pretty much the same. It's just less confusing to worry about this whole ratio thing.
Yeah, and I want to address some comments. I can't use different graphs. This is the actual data. You can click on this paper in the description below, and this is the exact data the authors used. The goal of my streams is not necessarily to teach you facts. It's to walk you through and help you think about data. So sorry to say, I will be using only the data that I am given. These graphs, if they are bad, they are bad. But also, if you go to graduate school, you will be thrown so many difficult graphs that you'll get really good at figuring them out. For example, I've not seen any of these graphs, and given I didn't do a perfect job the first time I read them, but part of what is so rewarding about being a scientist and learning to think like a scientist is challenging yourself to think deeply and to interpret data that can look very difficult to interpret. I don't know about you, Dr. Wendell, but my strategy for these is, okay, this graph looks complicated. I've never seen this graph in my entire life. I first take a look at what the authors want me to see, and then I try to see that. And then after I try to see what the authors want me to see, then I can critically think whether or not I agree with them or not. But yeah, I agree, it's not easy. If it breaks your brain, that's to be expected. Of course, this is the equivalent of graduate school. So if you're having a hard time following along, just know that that is understandable. But if you can follow along and engage with the discussion parts, that's also equally valuable. All right. So let's move on. This is another violin plot, but this is with the opposite chemical. So the first one was with dopamine. This is with ticlopride, which is a dopamine receptor agonist. Agonist or antagonist? I may have gotten this wrong. Antagonist. I used the wrong word. So this is a molecule that blocks dopamine. So this is sort of what happens when you stop dopamine from working. And what's interesting about this graph is that it doesn't seem to change overall neuronal responses to surprising sounds. So if we go back, dopamine reduces the overall activity of neurons in response to surprise. Blocking dopamine doesn't seem to do the opposite, which I think is worth noting, although I don't have the APM in my brain to draw any further conclusion with that.
Yeah, do you mind if, because I was thinking about this a little earlier too.
Yeah, absolutely. Go ahead.
I think something important to think about here to explain what might be going on here, and it's not so clear why this would be different, is if you think about what you're measuring with adding the dopamine, right? Like you're dumping a whole bunch of exogenous dopamine onto these cells in a way that's not really commensurate with
exogenous for my audience. Exogenous means from the outside. So dumping dopamine from outside of the brain to inside the brain. Yep.
Right. Which is not how synapses and neurons work, right? Like it's like small little spurts in like pretty specific areas on other neurons is like kind of how that goes. And so this, like adding this dopamine antagonist, it's not really just the opposite situation of adding the dopamine, because really now what you're looking at is like, you have to think about the amount of dopamine that is coming from inside the brain. Like that's what the etaclopride is blocking in this situation. So it's probably much less and in much more specific areas. So they're not really analogous situations. It's as close as I think you're going to get. But, if we're talking about controls and kind of like this is a control in and of itself in a way, it's not the same sort of experiment that you're doing just because in one situation you have an agonist and in one situation you have an antagonist.
Yeah, so I'm wondering if we could come back to this particular figure later when we kind of put it all together, because to me, I struggled to understand the significance of what this was meant to tell me. But maybe we'll revisit this. Unless you think this is important enough to hammer out right now.
No, I don't necessarily. I'm not sure that I know exactly what the significance is of this either.
Yeah, hard paper.
I have some guesses, but they are just pure guesses.
Okay. So maybe we'll move on from this because I don't see the obvious reason why this was included. Okay. So dopamine blocker does change things on a neuron basis. So again, this is the same neuron by neuron analysis of looking at the response to the deviant tone versus the standard tone. And instead of dopamine, we have the dopamine blocker. And again, we see this two-level different response to the shocking sound, right? You have some neurons that react more to the surprising sound, just like dopamine. And we have some neurons that respond less to the surprise, the ratio of the surprise, than dopamine. And what agonizes me here is that every one of these has the covariant of having liquid injected into the brain, which could explain that why there's this heterogeneity in both, that this neural trauma could be spatially dependent, that if you injected the neurons and the volume pushed up against the neurons in location A, that location A neurons might have this difference in reaction. So it's not a likely hypothesis, but because this is an effect that also happened in the dopamine part, it just, to me, it looks like the dopamine and the dopamine blocker kind of had the same effect on a neuron to neuron basis, even if it's not true at a population level, which is kind of bizarre. A asks if we can engineer mice to produce this blocker, and my answer to that would be it's kind of hard to get mammals to make small molecules. We're not very good at that, so to genetically engineer a mammal to do that would be potentially challenging. Okay. So they just said a control for the birds. I mean, they could have had a reason, but my brain is too small to figure that out. Never attribute an author's not getting the author. Like, the last thing you want to do is suspect that the authors are not good. First you doubt yourself, and then after you have made sure that you're not crazy, then you can doubt the authors. But I might be crazy, so.
Oh, actually, so I had a thought on this, Phi, and maybe you can explain if you have familiarity with this. So they use this thing called iontophoresis. Have you used that before?
I have not. I've only done volume injection.
Gotcha. Yeah. So I guess that was the thought that came up for me. Is so iontophoresis, as I roughly understand it, is you basically use an electric field to kind of like push the molecules out of a little pipette and into the brain. And I'm wondering if they don't have a vehicle control because the volume of injection, quote unquote, is basically just the volume of the particles of dopamine that they put in. And so how would you even do that?
But
with a saline solution? I don't know.
If there's not appreciable volume, then I don't know how far this dopamine can go, or if you know what I mean. I would. Okay, so I think basically what we can say is that we might have dropped the ball on fully understanding or whatever the heck it is. Iontophoresis. Yeah, that's not a method that I ever heard of before. So if there are methodological quirks with that as a choice for the experiment, maybe that could explain away why the vehicle injection didn't actually happen. It's pretty interesting. All right. So yeah, I mean, I could be eating my words later if any neuro people can figure that one out. Okay, so we did all this just kind of assuming that what we were measuring is a neuronal response to surprise. And here's another exciting part of chat getting to come back into the seat of the scientist. The authors wanted to make sure that everything that they had measured so far was in fact surprise and not simply change. So how might you experimentally distinguish change versus surprise? And I'll give you a hint. They're doing it with sounds again. So surprise, they were doing note, 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 and then a different pitch note. How would you do change and not surprise? Think about music. Any of you who are inclined to think about music. What would be a change in the musicality of something, but not a surprise? Change the notes slowly, says A. That is definitely true. Can we get a little more nuanced? What's the difference? A surprise is a change that you don't expect, but what's a change that you can expect? Halftone up or down? Yes. Scales? Yes. Change notes slowly over time? Yes, yes, yes. I was looking for halftone up or halftone down or scales. Some mathematically defined predictable change. Instead of note, 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 high-pitched note, you get whatever a scale is. I'm not a musical person at all. Isn't surprise a reaction to change? It's a type of change that you don't expect. So I don't know if you were here for the first half of the stream, Thomas, but we're basically saying that the brain predicts or your experience of reality is your brain predicting what should happen, and sort of your experience with the lived experience of your world is you adjusting to the error in your prediction. So a surprise is something that goes against the reality that you have predicted for yourself. Yes, so great job, chat. You distinguish change versus surprise by doing scales. So here is their figure, and I really love this. So you have on the left here, unpredictable changes. So you play the note, and then in their figure, they have a note that is lower in tone, and so that's their deviant tone. And on the right here, we have a change that is not surprising, going up or down, whatever half tone that is, but basically scales, right? So when you see DEV, that's the deviant tone, STD, that is the standard tone, and then CAS is the predictable change. Alrighty. So let's take a look at how dopamine works with response to change. So on the left here, we have the predictable change, the CAS, and we see that dopamine, it says NS, so it's not significant. So we can basically conclude that dopamine doesn't affect the neuron's firing rate in response to change. In the middle, we have the deviant tone, so that's good. That's replicating the same data that we saw before, that we have a decrease in firing rate. And then again, the standard tone on the right, they're not different at all. So the authors are pointing out that this type of learning is not happening just when things are changing, but when specifically your expectations are not met, or to use the language that should be the theme of the day, when your prediction is not accurate. Okay, so putting all of this information together, the classical models of cognition would suggest that dopamine is released in response to making a correct prediction, right? Generally, we think of dopamine as the reward molecule. So generally, you're rewarded when you are right, and your brain kind of punishes you when you're not right. Classically, that's how we used to think of things. But experimentally, dopamine is also released in response to things that have no obvious reward or punishment associated with them, i.e., something that is just a surprise. So the goal of the paper is to discover what the role of dopamine in learning how to deal with surprises is. The way that the authors did this was to measure the neuronal response after dopamine, before and after dopamine is added in response to a surprising tone. And their results, dopamine decreases neuron activity in response to surprise overall. So overall, neurons are not as active in response to surprise. And maybe to tune exactly how much to quote unquote learn, that's just my thought. But it can decrease/increase activity depending on the individual neuron. So this is not a blanket change. It doesn't quench every neuron's activity. Some neurons get more active in response to surprise, some get less active in response to surprise. Dopamine blocker does not change neuron activity in response to surprise overall, so it's the presence of dopamine that is doing this change. And this response, importantly, to line it up with predictive processing, is only in reaction to surprise, when your expectations, when your predictions about the world happen to not line up with the sensory information that you are getting in. Change is a type of, incongruence, but it's not an incongruence that we can't predict, right? If you are playing a scale, you will be able to predict that next tone, assuming you're not tone deaf. Okay. So this is getting into what Dr. Wendell was saying in terms of assessing this paper overall. Dumping dopamine around neurons might not be the most representative experiment, because the dopamine is not coming from dopamine neurons. So if dopamine's not being released, like what a dopamine neuron would do, you don't quite have the same spatial release, so it's not really in the same places as where dopamine would release dopamine. Dopamine neuron would release dopamine, and it's not quite in the same temporal situation, right? So neurons dump their dopamine in packets. Those packets can cause these pulses of signaling, but if you just dump dopamine, everything's getting dopamine all at once. So there's some caveats here, and I'm wondering what chat thinks about these caveats and how severe these caveats are for coming to this conclusion. So, are these caveats enough to quote unquote sync this paper for you? And remember, the thing that we've concluded is that dopamine reduces, I think, okay, and Dr. Winslow can correct me if I'm wrong, but the main takeaway that I had was that dopamine causes neurons to be less responsive to surprise. That's, I think, the headline of the paper. Would you agree, Dr. Wendell?
Yeah, no, I think that is the headline of this paper.
Thomas has a hypothesis. Tell me if my hypothesis is stupid, but maybe dopamine is released in response to surprise as a reward, so animals seek out more learning opportunities and gain more knowledge in general. Dr. Wendell, do you want to talk a little bit about that now that we have this space?
Sure. Yeah. So I would offer a different sort of way of thinking about this, trying to get away from this idea of dopamine being kind of a reward molecule. If we think about, like, in the context of just this experiment, right? What have we seen that dopamine does in the context of this experiment? Well, it seems like it dampens the error signaling from neurons, right? So I might hypothesize then that actually what dopamine, when dopamine gets released in situations where our top-down model is saying, the error signals from this stream of data, not that important actually. Right now, I'm focused on my visual pathway. I don't really care so much about what I'm hearing. I'm trying to figure out what I'm looking at right now. So I'm going to release some dopamine into my auditory pathway, and I'm going to dampen down those error signals because they're not that important right now. I think that's what this experiment offers, is that way of thinking about dopamine as a way to modulate attention, like we talked about earlier, or concentration or focus, by quieting down less important parts of our sensorium, of our sensory information.
Yeah, and in terms of having the evolutionary hypothesis, I think the gist of, or the energy of the paper, I'm assuming, is that we can separate reward from this kind of neuronal adjustment. That it's not that you're wrong, Thomas. It's more that we've been stuck, I think, in this, dopamine is the reward thing. But it might be helpful to think of dopamine as also having completely not reward-based function, just like pure neuron adjustment kind of stuff, right? Because, naively, from my perspective, now that I have finished the paper and talked to Dr. Wendell extensively, I am now seeing the utility of trying to separate myself from believing that dopamine is dogmatically only the reward movement molecule. Like, it can be just active in a context of adjustment of prediction. It doesn't have to have reward as a part of its story. It might, Thomas, you could be right, but we don't have to think of dopamine as only being something that drives reward. So it seems that most of you are in agreement that this caveat is not enough to sync the paper for you. And I agree. Like, it might have a pretty artificial setup, but they did it in a brain in a living mouse, right? This wasn't neurons in a dish, right? This was a living mouse. Sure, could it have been cleaner experimentally? Yeah, I don't know how you would. So let's think. That's it's a good question here. So chat. These caveats, can we think of an experiment to do where these caveats no longer play a role? Can we fix these caveats? And again, for my audience, I know that you are not neurobiologists, or most of you from the poll that I took at the beginning of stream, but suggest something that you could try, and then I can tell you, or we can tell you from our experience, whether or not those are possible. Give the mouse a shower to clean up. That would make the mouse cleaner, although they do a fine job of grooming themselves. So dopamine, we dumped it into the brain. What could we maybe try? And again, don't worry about whether or not it's possible. Pushing yourself to come up with creative solutions is the mark of a growing scientist. get a mouse neuron to release a dopamine. So that is interesting. I want to hear, that is what I thought too, Thomas. I want to hear Dr. Wendell's thoughts on whether or not we could do this, but instead of injecting dopamine or ionoelectrophoreting or whatever into the area, whether or not we can stick because I know that you can. I know that you can stick a single electrode into a neuron and force it to dump neurotransmitter. I've seen it done before. Whether or not, Dr. Wendell, do you think that that is an experimental design worth pursuing? Because I haven't thought too deeply about this. It's just the thing that my brain first went to.
Yeah, no, it was the thing where it was where my brain went to too. I think it's a catch-22 in this situation, right? Because you can say, well, dumping a whole bunch of dopamine isn't really like how this would actually happen. But I think you could say the same thing about getting the mouse neurons to dump their own set of dopamine. Like, that's also not how that would happen. And how selective can you be? Then I started thinking about, there's this really cool, and I know, I'm sure, Fi, that you know about this, but for the edification of your audience, there is optogenetics, where you can have certain neurons express, be genetically modified to express light-sensitive proteins. And then you can stimulate the neuron by using a light source in the brain to get them to just the neurons that you want, essentially, if you can target your population well enough to fire those. So, I mean, it might be, I don't know if it would be cleaner, but it would definitely be different, and it would give us some more information about how these results look with those two different experimental designs.
I want to address Hairday's question. Could you measure where the dopamine is coming from and its amounts? So I'm not as familiar with dopamine because calcium signaling is for glutamatergic activity, right? I don't. It's been a while since I've done that kind of thing.
No, well, it's complicated, as you know, Fi.
Yeah, it's complicated. But I'll say this in response to Hairday. I want to challenge you to think a little deeper about whether or not you could generate a hypothesis from that. So you could certainly do that, but I want to challenge you to think about why you would do that or what kind of data you would generate. Is there a way to remove or inhibit dopamine production to see the differences? So they dumped in the dopamine blocker as a part of their experiment here. I don't really know if there's more to that you wanted, Aeon. Do it in a petri dish outside the mouse. If you do it in a petri dish, you remove the organism level of surprise, right? Like neurons in a dish, what surprises them is really hard to know. Are they even capable of surprise, right? You start to go backwards in terms of your validity or face validity.
And then I think, I was going to get at what Aeon's saying. I think, Aeon, maybe you're asking, what if you could just do this with, what if you could just modify how much dopamine a mouse produces generally? That runs into a whole bunch of, dopamine's an incredibly important neurotransmitter throughout the body and the brain. And so you'd have a lot of questions about, well, what is tweaking dopamine globally doing? Does it really look like a mouse or a rat at that point?
And correct me if I'm wrong, too much dopamine is the etiological agent of Parkinson's, or not enough dopamine?
Not enough in a very specific pathway. Yeah.
I see. It's not a bad idea, Aeon. I think that kind of thinking would be okay in a lot of other biological systems, but the brain is, it's tough. It's tough. Good thoughts, y'all. Good thoughts. Very, very cool thoughts. Love that. Okay. So this is kind of a little plot to get a general idea, I think, of what we've learned. You have a surprise that causes dopamine to be released, And then that dopamine can either sensitize or desensitize neurons to fire differently in response to that surprise. Overall, it is lowering the firing rate, and that somehow results in your neurons learning. Maybe not necessarily your brain learning, but your nervous system adjusting. I think now that is the appropriate amount of nuance I think that I've gotten from our conversation. Okay, so I want to give the floor to Dr. Wendell now to talk about this in the field of psychiatry. And I believe you've also written about this in your Substack, which is in the description below. If you've ever wanted to follow a psychiatrist's thoughts on how psychiatry is done, please go check out that Substack, because it's actually super fascinating. And I actually used his Substack article to prep for stream today, so that's been really, really helpful. And in that Substack article is a bit of musing as to how this idea of predictive processing relates back to psychiatric disorders, which is really cool. So Dr. Wendell, you have the floor.
Yeah. So I actually, Fi, you got a little bit of a sneak peek of that article. I still haven't finished writing it up yet. But it will be there soon. So yeah, I mean, part of the reason why, as I talked about at the beginning, I find predictive processing so interesting is because it might start to offer answers for why some of the changes that we kind of know exist in certain disorders, psychiatric disorders, produce the sort of symptoms that we see. So, for example, schizophrenia is treated with a class of medications that block dopamine. That's like what they do. And it seems to improve the hallucinations and the delusions in schizophrenia. It doesn't seem to help so much with the other symptoms, but with those symptoms, it certainly does. And so something like this experiment and this idea of predictive processing, I think, offers a really attractive explanation for why changes and abnormalities in dopamine signaling might produce things like hallucinations and delusions. There are different schools of thoughts within predictive processing about where the problem might be. But you can imagine if dopamine's job, right, is to help the brain filter out irrelevant stimuli, or assign some sort of importance or an idea about precision to incoming signals, you might see how, let's say, too much dopamine quiets down the sensory information that you're getting too much. And so you start to get your top-down models that the signal they're getting essentially, right, is like, oh, we're predicting really well, actually. There's very little error that is coming up from the bottom of the system. So we're good. In fact, maybe we should make this prediction even stronger and apply it to a whole bunch of different situations.
Oh.
And so, yeah, sorry, go ahead, Fi.
No, I'm just reacting. That's super cool. So I'm going to write this out in chat for a second while you continue. I think, to me, my previous understanding of the link between dopamine and hallucination was very different. And of course, I'm not a psychiatrist, so I had basically zero, but I had hypotheses, right? So my hypothesis was that too much dopamine was inappropriately reinforcing conceptions of reality. So like top down, like too much dopamine is rewarding you for thinking things that are not real. When what you are saying is that there's this disconnect between the senses and the prediction machine. And if the prediction machine can't get enough signal from the senses, then it starts to not be able to connect with reality as much, which I think is a very elegant way of explaining hallucinations. Of course, assuming that what I have said is correct.
Yeah, right. So that's definitely one conception of it. There's another conception that says, all right, like it's kind of hard for us to know, like there are, if you look at different pathways of dopamine signaling in the brain, there are, I think, 5 or 6 major ones that we could talk about. And in one of those pathways, dopamine signaling seems to be too high. But in another one of those pathways, dopamine signaling seems to be too low. And so if we take a step back and we just say like, all right, maybe we're not going to make like specific comments about dopamine being too high or too low in any particular sort of area, but we're going to say that dopamine transmission is just screwed up. And so error prediction is screwed up, like globally throughout the brain. And so there's a lot of uncertainty. And so maybe the delusions and the hallucinations that we see in schizophrenia is actually a product of too much uncertainty. The top-down models can't find a good predictive engine, a predictive model, because of how noisy and how much your bottom-up signaling is changing, maybe almost like at random. And so the delusions and the hallucinations that the top-down models kind of end up being attracted to, maybe those are the ones that just best account for this very noisy, sort of uncertain set of experiences. I think that this is compelling too if you think about what kinds of delusions and hallucinations people with schizophrenia tend to have. You know, like a joke I like to make is nobody with schizophrenia, ever suspects that their parents have been replaced with better parents. It's always that they've been replaced by, evil individuals. The FBI is after them. These are all kind of hypotheses about the world that move towards explaining uncertainty in a way that I think even evolutionarily makes a lot of sense, because they're all about being very hyper aware of danger and of threats. And if you're in a very uncertain environment, you're going to want to tune your top-down models to protect you from those things. You're not going to want to tune your top-down models to be expecting rainbows and butterflies all of the time.
That's really interesting. Although I'm sure, again, you can correct me if this is pop science, but I did hear that schizophrenia hallucinations are culture dependent and that there are cultures where the voices are not antagonistic. So I'm wondering whether or not that is explainable using predictive processing as a model.
Yeah, sure. I should say that even, here in the US, there are individuals who tell me that their auditory hallucinations are neutral, or that they have a mix of positive and negative voices. But I think I would be really surprised if there were places where it was totally culture dependent. Maybe there is, and that would be fascinating in and of itself. But certainly I think the content of the delusions, obviously the content of the delusions have to do with the cultural milieu you're in, right? Like nobody was hallucinating about the FBI being after them, 500 years ago before the FBI existed. But yeah.
I really like the idea. Again, not like I like how satisfying the model is for explaining away perceptions that are not reality, right? I think this idea that we are constantly living in a prediction mode, and then things that break, or things that, because to these people that are experiencing these delusions, that is their reality. That is what their prediction engine is generating. And this idea that dopamine has the potential to alter the error that is passed upward, and then thus breaking the machine learning, I guess, of the brain to create these delusions is really, really fascinating. I think that makes more sense to me than anything else that I've read about schizophrenia and delusion, I guess. Like, not that I've read that deeply, but like, to me, that sounds very plausible. I hesitate to say that is correct, because who knows, but as a way to generate hypotheses, I think that can be compelling. Although I'm not smart enough to know how to generate a hypothesis from that. But the fact that we have aspects of this idea that are testable, I think are really interesting.
Right. Yeah. And I think that also the thing that's so compelling too is that we don't need like crazy breakdowns in, like we don't need a piece of the brain missing. We don't need like, giant changes in differences between kind of the base models that we're talking about, right? Because we all have had the experience of daydreaming, or of hearing something that like we didn't actually hear. You can kind of reliably induce people to hallucinate, even in pretty simple experimental settings. So it's not as though the brains of people with schizophrenia are like crazy different and just have some like totally separate base neurobiology. Predictive processing just says no, like it's all the same like schema. It's just that here there are a few things going wrong in some of these bigger picture signaling cascades, and that's what gets them to this point.
I think that's also satisfying in the sense where it kind of kills the idea that there will be a simple biological etiological agent that causes schizophrenia, right? Like what I understand from my half of schizophrenia research, like my very small basic biology understanding, is that we don't know what causes schizophrenia because it's likely a whole bunch of different, very small changes that could lead you susceptible to just your wiring being just like a little different than everybody else's. And so the fact that there are so many immune proteins that are associated genetically with inheritance of schizophrenia means that neuroimmunology is crazy, because not only is the nervous system stupidly complicated, so is the immune system, right? So you have two of the most complex, annoying systems. And I think the key to understanding schizophrenia is to get a handle on both of these stupid, complicated systems, which is probably why we don't know what causes schizophrenia yet. Although, again, genetic correlates do exist. It's hard to actually pin it down, because if a whole bunch of tiny changes causes your dopamine to be just like a little different, that might be enough for all the wiring. Not the wire, I shouldn't say the wiring, because the whole point of it's not really the wiring, but the signaling might be screwed up enough to have that be an issue.
Yeah, well, it's probably both, right? Because you get long and even short-term changes in the wiring, depending on what the signaling is like. I don't want to downplay that idea, that there aren't real structural changes that may be going on, but I think to maybe a different degree, that's not the only place we should be thinking about. And to your point, like with the neuroimmunology, I don't think it's a mistake that all of these proteins like probably have a lot to do with synaptic pruning and all of that. It probably speaks to why why you see these aberrations in dopamine signaling to begin with.
Very cool, very cool. Aeon's actually had auditory and visual hallucinations from sleep deprivation basic training. That's also interesting. I don't know if you know off the top of your head if whether or not, because just seeing how broadly applicable this model is, just basically academic shit talking. Like so don't this is not our focus today, but do you know if dopamine signaling is changed during sleep deprivation, if that also feeds into this model?
I mean, I have no idea. I imagine it must be, because I think it's all of the perceptual stuff. Predictive processing demands that there is a way to adjust error, sensory error, and so if dopamine is involved in it must be. But how and why is a fascinating question.
Yeah, just wondering how. Oh, here's another question that is tangentially related. Are all hallucinations, like hallucination disorders, do they all have dopamine as an etiological agent, or?
So
it's all right if you don't know off the top of your head.
No. Well, I'm trying to think of because I don't know the exact answer to that question, but I do know definitively that the answer to that is no, or at least it doesn't seem like that is purely what's going on. If you think of the example I use is the classic psychedelics, like, well, we won't talk about LSD because LSD has some dopaminergic properties, but psilocybin is pretty much the magic mushroom. Psilocybin is pretty much just acting on the serotonin system. And that absolutely produces hallucinations and delusions. Now, if you think about something like serotonin as a neuromodulator, is it neuromodulating dopamine at some point in this chain? Maybe, but I don't think dopamine's not the whole story. Dopamine is probably just one of many neuromodulators that all have the ability, if they're out of whack, to cause changes in error signaling. And if you can screw with error signaling enough, you can screw with, our experience of reality.
That is really interesting. It's interesting that psilocybin is cleanly just serotonin, at least initially. That's very
right.
cool.
Yeah. I mean, it's going to affect, it's so hard to talk about the brain, any of these systems in isolation. I mean,
it's not clean.
Right, exactly. And it's a small, all of these, it's worth maybe mentioning that I think about 90% of the neurons in the brain are glutaminergic neurons. So they're excitatory neurons. And then the vast majority of the 10 or 15% left over are GABAergic inhibitory neurons. And then a tiny percent are dopamine, serotonin, norepinephrine, really, really small. So we probably, there's really no way to talk about one without talking about all of the others, because they all modulate each other.
Yeah, that's biology for you. Everything's kind of messy. So I have another question, and it's another bit that I had read in your Substack, but do you have a hypothesis for predictive processing in something like ADHD?
Yeah. Well, so I think that this story of dopamine as modulating, this is kind of we talk about dopamine and attention, right? Because how do we treat ADHD? We treat ADHD with stimulants like Adderall or Ritalin cause more dopamine and more norepinephrine to be released in the brain. It makes those things more available outside of the cells, right? And so if dopamine's job, at least in some pathways, some sensory pathways, like we see in this experiment here, is to quiet down those pathways and make it easier for the brain to say, oh no, that's not that important right now. This just makes perfect sense, right? It explains why dopamine helps people to focus more. It's because dopamine is literally quieting down, distracting, unimportant error signals that are coming from other sensory pathways.
Yeah, I'd have to say, just naively, the way that you explain, because I feel like without concrete application, it's very hard to get a handle for predictive processing. But when you couch it in terms of ADHD and schizophrenia, that illustrates what I think the authors are trying to get across. It didn't click to me until I read your bit about schizophrenia and ADHD, the context of error, and the importance of error and your perception of reality or focus, or just to me that seems very fundamental and deep. And I don't actually know how quote unquote popular this idea of predictive processing is. Do you have a general sense of this as a fringe belief, or if it's up and coming or dominant? This is the first time that I've read about any of this.
I would say that it's not fringe. It's an up and coming framework. It's becoming more and more popular. I'm trying to think. I don't know when the seminal papers for this idea was started. There's this free energy principle. Carl Friston is kind of the grandfather of this field, and I think his stuff started to come out 90s probably, but it's taken a while for it to really start to be fleshed out. And I'm seeing it in more and more places now, and more kind of big journals.
Yeah, I maybe it's because science is also human, right? Maybe it's because the classical models have better PR because it's just easier to understand.
Yeah, maybe. And I think understandably, people are kind of, they're always suspicious about big new theories of everything. Yeah.
So I have a question from Thomas to go off of our discussion ADHD, who's asking, what exactly is ADHD? I'm guessing asking about a mechanism of some kind. Do you have any insight into that? Because they have it.
Yeah, well, same. It's a good question. I don't really know. We do think that there's some sort of dopamine imbalance going on in ADHD, but again, it's a collection of symptoms, right? It's some difficulty with attention and impulsivity. Uh, I think it's kind of better to talk about it as maybe like an executive function sort of disorder, but it's not a place that I've spent a lot of time like kind of learning the underlying mechanisms because, you know, truth be told, it's not always terribly useful to do so in psychiatry. Sometimes it makes things more confusing than less confusing to really try and understand the neurobiology. Um, so
Can I ask you a bit of a personal question? It's okay if you don't feel comfortable with, but I think it would provide
yeah,
value
shoot.
to people listening. But like, you know, you have ADHD, but you're also a full ass doctor.
Yeah,
ADHD might have a component where it hampers executive function. Can you give us a little insight as to how you made it to such a high achieving, you know, very demanding kind of job?
sure. Uh, I always have a hard time talking about this because I don't want to sound like I'm bragging or anything when I do. So part of it was I'm very fortunate that naturally I'm smart, and that got me a very long way. I was diagnosed in middle school, and I got put on drugs. I got put on way too high of a dose, and I hated it, and I wasn't having problems in school. So, you know, I just kind of got through. But then, I mean, I crashed and burned really in my first year of college because the demands were so high for my attention and my focus, and I had never learned how to deal with that. I wasn't on medication, and I'd never learned how to study in high school because I didn't need to. So I got on stimulants, and I learned how to study. And I kind of, I think I had my first semester, I had like a 2.8 or something, and I graduated with like a 3.5, which I was, thrilled about. But yeah, so, stimulants and learning how to study and how to focus. And then I think also a big thing that I had to kind of deal with was like, I need to work and do things that I find intrinsically interesting because if I don't, I'm never going to stick with it. And so, I am lucky that one of the things I found really interesting is neurobiology and psychiatry. And so I love it and I find it fascinating and it helps. But yeah, it's been tricky. I think I've been very lucky in a number of different ways, though, in terms of how bad this disease could have been for me. I think I won quite a few cosmic lotteries on the way here.
Well, I guess it's probably not controversial to call it advice from a psychiatrist to stay on your medication or to make sure that your medication is working for you.
Do what your psychiatrist tells you to do, friends.
Yeah.
I mean, or at least talk to them about it.
Great. Yeah, that's super interesting. I find this is something to jump off of the like needing the stimulation of something as high achieving as getting into neurobiology. But my advisor used to say that she suspects that many people in the academic sciences are neuroatypical because you might have to be to to fixate on stuff enough to actually make meaningful progress. It was basically, the same thing of academic shit talking, but like, it makes me think about it sometimes where like, I don't know where on the spectrum of normal I personally am. I've never been evaluated for anything, but like what I do know is that if you choose academia, you are choosing to make your life your work and your work your life. And I feel like that's not an easy decision to do unless you deeply care for it, whether or not that care is wired in you or from your heart or whatever, however you want to conceptualize it. It always makes me think about that kind of idea. There's some interesting questions for you. What does predictive processing have to say about hyperfixation? And I think we kind of went over this already, right, in ADHD? Or what about OCD? Oh, we didn't talk about OCD yet.
We didn't talk about OCD. Well, I think the last one's question about hyperfixation is kind of interesting, because it seems it's kind of a counterintuitive sort of idea with ADHD, where the idea, like people can't focus, they bounce off the walls, blah, blah, blah. It's interesting. I mean, I think predictive processing, if you frame ADHD as a problem with, to some degree, dopamine modulation of attention, there's no reason to think that it can't go in both directions, where you have problems with dampening in some situations, and then in other situations you have, overfixation in other situations. That is a bit hand wavy. I will cop to that, but it's hand wavy because I don't have a more concrete explanation for you. But I think it makes sense if you're going to think about it in that sort of context.
I think you have
problems with.
I think you have experimental support, because we've already seen that the presence of dopamine can modulate neuronal behavior up or down in response to surprise at the very least. So there is heterogeneity, and I'm really wondering which neurons are which neurons have their response sensitized and desensitized, because that kind of gets to what you're saying about OCD, right? Like some could be up, some could be down via dopamine, and really wondering if that's like architecture based, like location based, or whether or not there's some more complex determinant of which neurons are wigging out and which ones aren't. It's pretty fun.
Yeah, it is. And there are, there are papers on this that I haven't spent a lot of time reading, but like that talk about like there are subpopulations of these different areas that are dedicated to doing different things. Some of them are providing crosstalk between different neurons. Some of them, are responsible for taking in the top-down inputs, so on and so forth. So it makes perfect sense that you have differential responses to dopamine, because you almost certainly have 3, 4, or 5 different subpopulations of neurons in any given area of the cortex that just have different processing roles.
This is why I liked my corner of neuroimmunology, because again, we were just doing neurons as funny cells, like all this complex interaction between circuits and what it means for the organism is a lot more than I was willing to think about in graduate school. But it's really cool. I'm firmly, because you had sold this idea to me, and I was like, oh yeah, that kind of seems cool. After talking about it and after putting myself through this paper, I'm firmly in the camp of like, this is some cool shit. this is very cool. I kind of get it now, and it has a lot of explanatory power, and it's really, and again, I keep on having to say this because I don't want to make it seem like, oh, I believe this full-throatedly. There's not a counter argument, that I'm aware of. But as a model that has high potential for explanations, that's very satisfying. Like the mystery behind delusion and hallucination to me is somewhat more solved, although solve is maybe too heavy a word. It's explainable in a way. There is a potential explanation, whereas before it was just like, I don't know, dopamine up and then you do weird brain things. And just having a starting point where you can test the hypothesis is very neat.
Mm-hmm.
As someone with ADHD, my OCD symptoms get worse whenever I'm on stimulants, which is interesting. That would... Yeah, I mean, I guess that's also just like, we're just talking into the void right now because of how, I'm guessing, new this application of predictive processing, and it is an interesting thing to note, but I don't think we can say anything further than that, especially when we're talking about a real human, right? Like, we can talk about, between the two of us on stream, we can talk about our particular medical whatever. I think I draw the line at talking in depth about anyone else's lived experience on stream, even with a licensed psychiatrist, just if I seem cagey.
Yeah. No, I mean, abstractly, though, it would make sense. OCD is kind of its own interesting thing that kind of, is it an anxiety disorder? Sometimes it looks even a little bit psychotic, right, with people with really severe OCD who have beliefs that, like if they don't do their counting, their family will die, that sort of stuff, which I mean, sounds quite delusional, but doesn't quite look like schizophrenia. So, though interestingly enough, I mean, the medications we use to modulate OCD or to treat OCD are the antidepressants, more than, the antipsychotics. So, maybe therein lies some sort of a clue as to why they're different. But serotonin is its own, maybe that's a journal club for another day, serotonin and predictive processing.
Oh, God. Not for a while. My brain is processed out. My synapses are exhausted. There are no more neurotransmitters left. Okay, so we are pretty much going to wrap up the stream. I kind of want to give you, if you have the time and if you have the presence to maybe, because I know a lot of my audience are interested in health as a career, and I don't know if you would mind sharing a little bit about your journey or sort of advice or something, just to maybe round this out as a psychiatrist, right? So like my whole thing is, I love biology. I think biology is accessible for anybody, and I use biology as a vehicle to understand science. So my primary thing isn't to teach biology, but it's to teach how to think using biology. But because of that, I don't really have the opportunity, because I don't have career advice for anybody. My job is pretty unique where I can't give anybody advice. But since there are a lot of motivated biology people who might be thinking about a career in medicine one day, is there any parting words of wisdom that you would like to convey to those of them who are interested?
Yeah, so I'm not someone who's very gloom and doom about medicine. I think it's a wonderful profession. I think that it's fulfilling and offers fulfillment in a way that not a lot of other jobs do, personally. I think maybe the most appealing thing for me is that when I was trying to figure out what I wanted to do with my degree, it looked to me, it was like basically like go work in a lab, go do consulting, or go into medicine. And medicine was just the easiest choice because it preserves, or it allows me to interact with people. And at the end of the day, like the work that I do, I see as kind of being in service of others. And, realistically, the person who goes and works in a lab and discovers some, very amazing drug will do probably infinitely more for others than I will be able to, if you look at it from a mathematical point of view. But it's really important to me to be able to see like what I do be reflected in the people that I help and that I serve. So, don't, if you think that kind of stuff is important to you, think about medicine. It can be grueling and difficult, but I think it's worth it at the end of the day. Don't go into medicine if someone else wants you to go into medicine. Those are the most miserable doctors.
I think Dr. Wendell had started by saying that he's not all gloom and doom, because he's seen my stream, The Psychic, and I am doom and gloom.
Yeah. And I get it. And I think you're right to have people really take a good hard look at whether or not they're the kind of person that can hack it in medicine, because I think you can want like all of the things about medicine that I've talked about, but know that like realistically you just can't. You don't want that kind of life. You don't want those sorts of hours. You don't want that kind of responsibility. You can't, hack that amount of work. And it is important to be honest about it, but I think for the people who can do that, it is really an amazing it's an amazing profession.
Yeah, and you and Psychic are both proof. You and Psychic are both proof that there are happy doctors who are fulfilled. So even though I'm doom and gloom, it's for some people. Thomas wants to know why you want, or when did you know that you wanted to do psychiatry? I think that's an interesting question.
I think maybe junior or senior year of college. When I figured out I wanted to go to medical school, it was like, and I'm going to medical school to become a psychiatrist. That was really like, I knew I love neurobiology. I still love neurobiology. this has reminded me how much I love it. So thank you, Phi, for reminding me of how just damn cool neurobio is. So cool. But I wanted to work with people, and I wanted to get to know them over a long period of time. And also, there's a little bit of me that's kind of voyeuristic is the only way that I can put it. And I don't, it sounds a little sleazy. It feels a little sleazy, but getting to know people on a really deep level is just, it's a privilege in a way that people let us into their lives the way that they do. And yeah, no, I mean, it's just, it's very meaningful. And I wanted to, that's what I just wanted to do. I knew that that was for me.
Yeah, and contrast that to someone like me who is very happy in a lab kind of environment where I don't get to see anybody. Or at least I was in graduate school. My job is obviously a lot more social now, but I think that kind of human component, you say it's voyeuristic, but it's also just like how people have functioned for thousands of years. Thousands of years. Right. It's like now we would consider that voyeuristic, but you know, it's very human, very natural, very cool.
Yes, well, there are a lot of uncomfortable, untoward feelings that are very human and natural, which is why I try not to shy away from calling it voyeuristic.
That's fair. That's fair.
Because it is, you know. I mean, but that doesn't mean that that you have to let it dictate your behavior, right?
Your relationship with your patients is also sort of unique, I think, in psychiatry, because you are in their brain with them in a kind of way. Like, not only are they with you, you have to figure them out too, like who they are. Occasionally, you know, in very severe cases, you are a part of the formation of self. And I think that's kind of, I don't know, that's kind of heavy. Do you feel that that's kind of heavy?
It is. No, I mean, it definitely, it is very heavy. And it's unique, I think, to psychiatry generally. You know, I mean, there are a lot of very, I don't want to diminish other specialties because some of them get treated as very kind of mechanical and bloodless. And, you know, I mean, you can practice medicine that way if you want. But there is something unique about psychiatry and the types of relationships that we form with our patients that you really just don't get elsewhere. You really don't.
I think what sold it to me, not that anything needed to be sold to me, but I've had another psychiatrist explain to me that, you know, this is a field where you can actually see change. Like, I mean, that's true of a lot of other specialties, right? Like, it's not like only psychiatry has the capacity to change a patient's life. That's ridiculous. But in the sense where you can return their like true being. And I don't know, there's something very poignant about that. I am jealous of that level of maybe spiritual importance of the practice of psychiatry. I don't know if you feel similarly, but of course, getting the body back, that's also very important, right? But to me, what is more emotional is this idea that you are restoring someone's personhood in certain cases, or full personhood. Or I don't know if that's problematic to say, but it's kind of how I think of it.
Mm-hmm. Yeah, no, I mean, I know what you mean. Certainly the transformations that some of our patients go through is like, you know, it's almost like they're totally different individuals before and after. And yeah, I mean, it can be incredibly meaningful for patients where you kind of give them back the ability to have a life and be themselves.
All right, I think it's a pretty good note to end on, y'all. I hope you all enjoyed the stream. Thank you, Dr. Wendell, for joining me and for helping me. I don't know, I think I've had 6 revisions or something of this PowerPoint because it was I had so many things that I got wrong. I've actually, a little peek behind the scenes, I've never felt this stumped by a paper since graduate school. Since legitimately the first year of graduate school, this is the most I've ever struggled with a single paper. And Dr. Wendell has been very patient and kind and correcting a lot of what I had gotten wrong. So even if it was like very hard on me, because Dr. Wendell had helped, I was able to put together something that I think is not embarrassing, that I think had value and interest, and I hope you all feel similarly, that thinking about the brain in this kind of way, it's not something that I think I will regularly do on this channel because it is pretty far from thinking about cells and molecules, but certainly getting to view the brain at the level of the cell is something that I'm interested in doing in the future, because that's the only way that I can conceptualize things. Anything more abstract than that is going to truly blow my brain up. It is very hard.
Yeah,
all right. So
it is.
thank you so much, Dr. Wendell. I'm actually going to hang out on stream just to chat a bit longer. So I will be in contact with you. But for now, this is goodbye to Dr. Wendell. Say bye, chat.
Bye, chat. Thanks for having me on, Fi. This was a ton of fun.
Yeah, thanks for coming on.
Yeah, absolutely.
Alrighty. So that was cool. I liked that. That was awesome. I think that gosh, I love getting to talk about science with another person. When it's just me, it can be kind of tough, you know. Especially when it comes to papers that are this complex and sort of out of my comfort level. But here's a little bit of a treat for everybody who's left in the call, in the stream. I have launched my own Discord channel, but I didn't want to do like a full kind of launch. I wanted to roll it out bit by bit, just so that you know, we don't roll it out to everybody and then find out we have some major security flaws. So as a little bit of a sneak peek, I have pasted here a Discord link where you can join our private Discord, where I can chat further about papers. I think I might do like trivia on Discord, maybe like a weekly trivia night, which would be kind of fun and not require a stream, or just any kind of content that I don't think actually belongs on the channel proper. I think it would be fun to do via Discord. I think that'd be very neat. You can also hear sort of my behind the scenes struggles with a lot of papers, get sneak peeks, stuff like that. Yeah. I think that would be cool to do, especially trivia night. I think trivia night would be very fun, because trivia actually lets me teach biology for real instead of some of the more abstract, difficult papers. And also getting to share what people find interesting is always very, very cool. Yeah, so we can use that link, and I'm not going to make it public yet until every step of verification is like fully done. So there's like a CAPTCHA thing, and if you can do the CAPTCHA and assign yourself roles, I will have considered that to be a successful Discord setup. Yeah. Is there anything else that chat wants to talk about with regards to this paper or anything in general? I'll probably stay on just for a couple minutes longer, just because I haven't streamed in a while. Missed talking to you all. I've been really excited to do this particular stream, just because it has shown me that it's kind of funny. I posted on Twitter, and by the way, if you don't follow me on Twitter, I'm at @NeutralPhil. I had this experience of like, I am very confident after having survived graduate school that I can teach myself anything. Of course, that's part of the membership incentives, is that I am that confident that I can teach myself anything. But, you know, that is a feeling and not an active thought that I have. Like, I don't think that I'm that smart, but I feel like I'm that smart, right? And I was severely checked by this paper, like, being made to feel not intelligent every once in a while is a very good humbling experience, because I was like, yeah, I've done neuroimmunology before, I'll be fine. And then every step of this process has been quite challenging. Welcome, Fire Ice. Just got here, but we'll watch playback. Yeah, it's a pretty interesting thing. I might do like a post stream, like text recap in the Discord, which might be like a neat way to engage with my community. And if you miss some streams, you might not have to, because I think, like, honestly, I don't know how much of my audience actually watches the VODs, because part of it is interactive, I don't know that it makes for like a good VOD watching experience. I think the live component is really interesting. So I think, of course, via YouTube, I would rather you watch the VOD, but for my analytics. But I think by having like a text channel in the Discord where I can do like a post-stream wind down summary, it could be a neat way to even paint out what do I want to call it? Not a repository, but what's a big what is it? big blanket thing, tapestry, to weave like a long tapestry of the information that we've learned together, which could be very cool. All right, so with that, have fun with the Discord link. I will see you all next time. It would be hilarious if the author of a paper you discussed showed up while you're talking about the paper. It would be pretty scary. I try my best not to like shit on anything or talk bad about the authors, because I'm willing to give them a lot of slack. My main complaints are usually things having to do with writing, because sometimes things are worded in confusing ways or written in confusing ways. But maybe one day we can do it together, chat. If we pump my numbers high enough, maybe one day I'll get one of these authors to show up, and that would be really funny. All right. Bye, y'all, chat. I don't know when the next stream is going to be. It might be a 101 thing next week. No strict plans for it. I just got back from my buddy's wedding, which was really fun. So I'm going to take a couple days and then figure out what to do for content in the future, all while putting together my actual PC. It'll make making this pre-on video a lot faster, so you can have that to look forward to. Imagine if the hypothetical maker of the Lactus video showed up. I'd rather not that happen. I don't want to imagine that, actually. All right. With that, I'm gonna call it for the day. Goodbye, y'all. Bye.